Preeti ma'am, may I start?
Hello, good morning. Good morning to everybody. Hello, good morning. It is myself, Dr. Mohammad Minhaj Alam, Mirza Ghalib College, Department of Botany, Magad University.
Good morning, sir.
Good morning.
Kindly mute yourselves, please.
Good morning, Anita ma'am. सरिता स्टार्ट करो, प्रिंसिपल मैम भी आ गई.
Okay. A very good afternoon to all. I, Sarita Agrawal, on the behalf of SS Khanna family, welcome you all in 2 days national workshop organized by Departments of Zoology and Botany of SS Khanna Girls Degree College, Prayagraj, on the topic Green Biotechnology and Therapeutic Potential of Medicinal Plants. So let's start this event with the blessings of God to make this event a successful one. As we know, medicinal plants have been used widely for the treatment of disease because of their broad therapeutic spectrum and no side effects. Their bioactive compounds can target certain viruses and can cure or prevent several viral diseases and infections. Recently, there has been a remarkable progress in the field of antiviral herbal therapy and drug development. Keeping all these things in our mind, We invite our speakers and organize this workshop especially for you so to make a proper communication about these antiviral drugs. Before we start the workshop with distinguished guests and vibrant participants, I would like to invite our principal ma'am, Professor Lalima Singh, to accord a formal welcome to our distinguished speakers and guests. Kindly join us, ma'am.
Sarika, can you hear me?
Yeah, ma'am.
Yes, ma'am.
Okay. A very good morning to all of you. And first of all, I would like to congratulate the Department of Botany and Zoology for initiating this process of organizing this national workshop on a topic which is of immense relevance today. The most valuable thing in life today for all of us are plants. This is one thing without which we cannot survive. Me being a sociologist and you all being scientists, the importance of plant for a scientist is of medicinal value. But we all are members of society, and therefore, being member of society, plants play another very important role in our life, and that is plants are worshipped by all of us. But since today, we are emphasizing on the medicinal value of plants, so we'll be focusing how these plants are now indispensable to all of us. Not only in India, in almost all countries, the medicinal plants have been used by one and all. If we just go through the data, we find that more than 80% of the population, especially even today in rural areas, all treatments are being done, all herbal treatments are there. In sociology, we talk about a concept that is called social change. And in that social
मैम काइंडली अनम्यूट योरसेल्फ मैम आप म्यूट हो गई हैं मैम अनम्यूट मैम म्यूट हो गया है आपका माइक काइंडली अनम्यूट
आवाज आ रही है?
यस मैम यस
हाँ
यस
मैम, अब
मैम
आ रही है.
So what I was saying, I was talking about it. Change is the law of nature, and being from the subject of sociology, I was saying that there are three types of social change, and one is the cyclical change, a change from when we return to the same point where we had started. Long years back ago, we were totally dependent on our health issues on plants. And now, perhaps COVID has again taken us back to all the herbal treatments which are being done by all of us. They are supposed to be the most safer ones, the ones who have less side effects. And as we go through the data, we find that more than 80% of the population today wishes, prefers, and takes herbal treatment first. This seminar, in which the speakers of the day, which is for a 2-day national workshop, we have eminent speakers, and I was going through the names. We have Dr. Anita Tomar, then we have Dr. Vishnu Agarwal ji is there, Dr. Navita Gupta ji is there from Jharkhand, We have Dr. Sanjay Singh, who's from Prayagraj, and we have Dr. Lal G. Singh, who's a scientist and head from Andaman and Nicobar Island. We are so fortunate that this seminar with the
Voice is not coming, madam. Your voice is not coming.
मैम सर मे भी नेटवर्क इशू है कुछ थोड़ा सा।
Dr. Sabnam, voice is not coming.
हाँ सर कुछ हमें लगा नेटवर्क इशू है कुछ, चेक करते हैं एक मिनट।
Okay.
आई थिंक प्रिंसिपल मैम लीव हो गई हैं।
डॉक्टर सरिता आप कंटिन्यू कर दीजिए।
Okay. Thank you, ma'am. Thank you for your guidance and encouragement for organizing such an event. Now we move on today's session. It is great honor for me to introduce and welcome eminent speakers in this session. Our first speaker is Dr. Anita Tomar, Scientist F in Forest Research Center for Eco Rehabilitation, Prayagraj. Dr. Tomar received Women Scientist Award in Second Rashtriya Vaigyanik Sammelan at Doon University, Dehradun in the year 2010. She was also adjudged for Brandeis Prize by Indian Forester in 2007 and recipient of Best Scientist Award in 2020 for outstanding contribution and recognition in the field of biodiversity conservation in 2021. She has published more than 100 papers, 3 books, and many articles in different national and international journals with high impact. We feel fortunate to have Dr. Anita Tomar among us as a speaker. Ma'am, I heartily welcome you in this workshop. Kindly join us. Anita, ma'am.
मैम अनीता मैम को कोर्स बना दीजिए, उनकी स्क्रीन शेयर नहीं हो रही है, उनको अलाउ करिए।
मैम एग्जिट हो गई है क्या?
नहीं है मैम।
मैम मुझे देख नहीं रही हैं। मैम एग्जिट हो गई हैं।
नहीं है मैम।
मैम है मैम, स्क्रीन शेयर हो रही, देखिए तो करिए।
नहीं, अभी को होस्ट नहीं बनी है।
अच्छा अच्छा,
मुझे वो दिख नहीं रही है। एक
हम लोग ट्राई करें, बना दें इसको।
हम लोग नहीं बना
आप
पाएंगे।
कोशिश करिए, हमें मैम का नाम ही नहीं दिख रहा है।
ऋचा टंडन मैम के नीचे है उनका नाम।
मैंने बना दिया है।
अनीता मैम चेक करिए।
now you can share your screen. You are co-host. Yes, ma'am. Share your.
यस मैम स्क्रीन शो नहीं हो रही आपकी, फिर से शेयर करिए स्क्रीन। अनीता मैम यस मैम विजिबल है स्क्रीन, स्टार्ट करें। मैम आपकी आवाज लेकिन नहीं आ रही है, स्क्रीन विजिबल है। मैम आपकी आवाज नहीं आ रही है। अनीता मैम आवाज नहीं आ रही है, आप म्यूट है क्या उसमें? माइक चेक। मैम आवाज नहीं आ रही है आपकी, आपकी वॉइस नहीं आ रही है।
Ma'am, you are not audible.
Yes, ma'am.
माइक वाले ऑप्शन पर जाइए और वहां पर जो सिलेक्ट माइक्रोफोन है वहां पर आप टैप करिए, उसके बाद फिर में भी आपकी वॉइस आ जाएगी।
अनीता मैम आप अपने को अनम्यूट करिए। अनीता मैम। अनीता मैम का माइक म्यूट है। शबनम।
शबनम मैम आप उनको फोन से इन्फॉर्म एक बार कह दीजिए।
Dear participants, kindly wait for some time. There is major network issue at Anita ma'am's side. Within 5 minutes, we have to start.
कैपेटिस जी रीच हो गई ना, थोड़ा उनको ज्वाइन में प्रॉब्लम हो रही है, वो ज्वाइन कर रही है बस 2 मिनट। मैम आ गई है, देखो सरिता। एक मिनट। अनीता मैम स्क्रीन शेयर करिए।
अभी को होस्ट नहीं है, को होस्ट बन
अच्छा।
जाए। प्रीति मैम को होस्ट बना दीजिए मैडम को।
मैम अनीता मैम को होस्ट बनाइए।
बना रहे हैं, उनका नाम इसमें स्क्रीन पे नहीं दिख रहा है। Am I audible, Shabnam?
यस मैम आ रही है।
यस मैम।
But you are
Screen
not
share
audible
करिए।
to me.
मैम स्क्रीन शेयर करिए, होस्ट हो गई।
अभी नहीं हुई है।
हो गई है, हो गई है।
यस मैम स्क्रीन दिख रही है।
हाँ विजिबल है मैम स्क्रीन। स्टार्ट करिए।
Can I start? Am I audible?
यस मैम
ये-
यू आर ऑडिबल। और स्टार्ट करिए। लेट स्टार्ट योर प्रेजेंटेशन। मैम स्टार्ट करिए।
आ रही है तो मैं स्टार्ट कर दूं। माइक में दिक्कत, तो मैं बोल रही हूँ, कोई किसी और को बोल दो ना कि वो रिप्लाई करे, अगर तुम्हारे उससे नहीं। मैम आप स्टार्ट करिए।
मैम लेट स्टार्ट। हाँ मैम स्क्रीन विजिबल है, आप स्टार्ट करिए। सरिता मैम से बोलो स्टार्ट करें। शायद मेरी आवाज नहीं जा रही है क्या?
शबनम आप उनको फोन से कह दीजिए कि उनकी आवाज आ रही है।
नहीं वो हाँ बोल रहे हैं। सरिता तुम बोलो, मैम स्टार्ट करें, आवाज आ रही है।
अनीता मैम, am I audible to you? अनीता मैम, हेलो।
मैम
Yeah, Dr. Tomar, you are audible. Just you start. Go ahead.
मैम स्टार्ट करिए। हाँ
शबनम आप फोन से कह दीजिए उनके ईयरफोन में कोई प्रॉब्लम है।
Dr. Tomar, you are audible. Go ahead.
इसको लड़की से लगा करके लॉगिन
अनीता मैम स्टार्ट करिए, आप ऑडिबल हैं।
Very good afternoon to all the person who have joined online today. First of all, congratulations to SS Khanna Government Degree College for organizing this 2 days national workshop. My warm regards to Professor and Principal Lalima Singh Madam, Dr. Sarita, Dr. Parveen, and all the students who are connected with me today. Very sorry for being delayed today. Maybe there is some problem and I'm not able to hear host anyone voice, even I am audible or not, I am not knowing. So if something is distracted, please call me in my mobile number. So the topic of my presentation today is on medicinal plants and their good agriculture practices. Today we will be talking about medicinal plant and overview in connection to India, threat to medicinal plant sector, categorization of medicinal plant into 7 classes for conservation, and good agriculture practices. When we start with medicinal plant, the one thing which comes to our mind that in our historical Vedas also there is written that no plant in this world is there which does not have medicinal property. Even if we have identified it or not, but all plants have medicinal property, and till date only some medicinal plant has been categorized that this is having this medicinal property. Some plants are used for medicinal purpose, for some barks are used, for some stem is used, for some seeds are used. Medicinal plants, we know that healing with medicinal plant is as old as mankind itself. Even the Indian holy book Vedas mention treatment with plants. Raising medicinal plants will help to use them effectively, and at the same time it enriches the biodiversity of plantation as well. We all know that India is the richest source of biodiversity in the world, and most of the forest area, most of the medicinal plants we get from the forestry sector. As I already told, the most of the medicinal plant we get from the forest, and only 10% of this are we got from open grassland, from agriculture pastures and other areas. Medicinal plant in Indian traditional system of medicine, it is one of the oldest system of medicinal practices in the world and has played an essential role in providing healthcare services to human civilization. In India, it has the exclusive distinction of its own recognized traditional medicine, whether it is Ayurveda, Yoga, Unani, Siddha, and homeopathy. When we talk about medicinal plant, an overview is that it provides healthcare to 80% of the population, and it constitutes world richest biodiversity. It provides raw material for traditional, alternative, and complementary medicine, which are largely plant-based. It is a source of 25% of modern drugs, and it provides livelihood to millions of people who depend upon collection, processing, and trading. As per the World Health Organization, the global market value for herbal industries is expected to reach, you can clearly see in this graph, it is expected to reach $5 trillion in 2050. And the global market growth for the industry is expected to be 7% per annum from year 2000 to 2050. Now, status of medicinal plant in India. India has 20 agroecological zones, and India is one of the 7 mega biodiversity country. It has 12% of biodiversity, including the medicinal plant. So it has a very rich source of medicinal plant. When we talk about the plants which are being used by various systems of medicine, this graph clearly show that how much the Ayurveda, Siddha, Unani, homeopathy, and Tibetan, and the modern system of medicine, the number of species which are used for this traditional system of medicines is shown in this graph. The folk healers in India, we can say that like for home remedies, there are in the first serial number, you can see there are several numbers, several million people who are using this traditional healer medicine. And herbal healers are 3 lakh, bone settlers are 60,000. So we can see how important and how in different categories the traditional folk healers in India are categorized. Now, distribution of medicinal plant by habitat. We can see the tree constitute 33%, the herbs 32%, and the medicinal plant in category of shrubs are only 20%, then the climbers are 12%, and others categorized into 3%. Breakup of medicinal plant by the parts utilized. As we say, there are many medicinal plants. Some we are using bark, some fruit, seeds, stem, leaves. So the whole parts we are using only 16%. The bark we are using from medicinal plants, our bark is only 14%. Then fruits 10%, seeds 7%. Flowers 5%, rhizomes, which are underground part, are only 4%. Now we have to categorize the medicinal plant, and here we have categorized the medicinal plant into 7 classes. Like restricted distribution, heavy pressure. The species here, the medicinal plant is species with a sparse population in few areas and which are in high demand. The second is restricted distribution, low pressure, the species with a sparse population, but at present low demand. So this way it is categorized into 7. The third is locally common heavy pressure. As we have seen in many, if we go to our rural areas in some villages, we can see some species are most commonly used by the local people. For example, when we go to Faizabad area, recently I visited that area. And there is one species, Stereospermum surveillances. Its root is used in Dasmula rest also. And the people of that area were using this species for migraine purpose, and they locally called this at Ardhakapari also. So locally common heavy pressure species with gregarious population in several localities, but having high demand. Then locally common and low pressure species which can withstand disturbances such as trampling and grazing and have low demand at present. Wide distribution, high pressure species widely distributed but also heavily exploited. Like we can see there are some species like during the COVID-19 scenes, we have seen the giloy, which we were using for Kara purpose. It was so widely distributed, but it was also heavily exploited by the people also, because some the 50% people was using the kara of this giloy, but some there were some numbers of people who were just collecting the material but not using it, so they were exploiting the planting material. Wide distribution, low pressure, widely occurring species, and at present low or no demand. Then under cultivation, low pressure, some species whose cultivation practices are not known. How, when, what is the correct time for collection of those species, and how we have to store the seed of that medicinal plant, that has not been known. So these were the 7 categories in which the medicinal plant we categorize. Then constraint in the development of medicinal plant sector. First of all, the constraint is inadequate financial support for commercial ventures. When we organize some trainings and the local farmers are coming, the one common they say कि हम लोग रेस तो कर लेंगे औषधि पौधा, पर उसको बेचेंगे कहाँ, और अगर हम लोग कुछ करना भी चाहते हैं तो हमें फाइनेंशियल सपोर्ट क्या गवर्नमेंट प्रोवाइड करेगी. So one constraint in the development of medicinal plant sector is inadequate financial support for commercial ventures, the documentation of local health tradition and practices, and one is the inadequate information on availability of selected species and resource management. If some farmer is interested to cultivate, for example, asparagus racemosus, the problem is from where he get the good planting material and what is the time of proper cultivation of that material and what is the time of sowing the seed. So these kind of information are not available to the farmers and other stakeholders. Then poor interest in the people regarding conservation issues and scope of sustainable development. And one of the most important factor is lack of awareness, lack of marketing avenue where the final material to be sold, and lack of scientific farming for authentic source. The threats to the medicinal plant species are slow growth rate, low population density, and narrow geographic ranges. This is the chart which clearly shows the name of the medicinal species and which parts are used and when the plant of that part which is used for the medicinal purposes is harvested. I will not take up the whole this table. For example, in case of we can say the bale, its fruits are harvested in the month of February, March, and the use is, for example, it is for use for cooling, diarrhea, and anti-diabetic. Say similarly, in case of the second serial number second, for example, in the Arjun, the bark is harvested, and when this bark is harvested, when the people are harvesting the bark, the whole of the tree is greatly damaged by the people. So here we will talk about the good agriculture practices when collecting the medicinal plant. So in, for example, 17 number, we can see Kalmig, Andrographis paniculata. Its entire plant is used. It is anti-malarial and liver tonic. Now coming to good agriculture practices, because while collecting the medicinal plants, we are destructing the planting material, severely damaging the planting material. So good agriculture practices of medicinal plants, it has been formulated to regulate production, ensure quality, and facilitate the standardization of herbal drug. Now coming, what to harvest? We should be sure, we should identify the plant that we need to harvest without any doubt. Means we should know, if we are going to the forest, we should know which plant we are going to harvest. The identification, the botanical service is important, because identification is important. We are going to collect some species and harvesting other species. It should not be the point. We should distinguish clearly between the medicinal plant and its closely related relative in order to avoid unwanted mixtures. Choose, when we are harvesting, we should choose the well-developed planting material, and don't harvest plant material that is infested with fungal growth or are having some poisonous byproducts. Be sure that the plants which we are harvesting, which we are collecting, are not sprayed with pesticide, herbicide, or fertilizers. When we are going into the forest for collection of medicinal plant, these problems don't turn up. But when we are collecting the medicinal plant from some farmers or other stakeholders' field, they have sprayed the medicinal plant with pesticide, herbicide. So we should be very careful during the collection purpose. Like in case of collection of underground parts, when we are collecting the planting material of medicinal plants whose underground plants are useful, the roots of annual plant must be dark when the plants are well developed. The plant should be well developed and it should be mature. Roots of perennial should be harvested late in the fall or early in the spring. Parts like roots and rhizome should be collected only after the seed shedding, and underground plants should be collected with minimum possible digging. For example, these are the ashwagandha roots, and this criteria, good agriculture practices, we should adopt. Like we can see, never use a sharp material for this digging the bulb or rhizome, which are underground parts. And whenever we are collecting the material, when we are digging this field, our instrument should not be very close to the roots, because it will damage the roots of a tree. So here we have seen, and after collection, we should better cover that area, cover that field back with the this potting mixture or the soil material, we should get it back at that place as it was before. Here we can see that it should have some, here in the first picture, we can see that the 30 centimeter distance से पहले से हमको dig करना है, and in the second picture we can clearly see we should not use such a sharp, like when we are collecting the underground parts, we are destructing the root of the tree. Now, it was the collection of underground roots. Now, collection of stem bark, like in case of Arjun, we collect the bark. Like in case of Albizia libic, also we collect the bark. So, stem bark should not be harvested when the tree is under new growth, like in spring season. The bark should be collected from mature branches of the tree, leaving the main trunk intact, and girdling of tree or branches by removing the bark all the way around should not be done. So these are the, whenever we are doing the stripping, we should have, not every year we should take up the same area of the tree where we have used the previous section. So it should be in an alternative, like a vertical stripping. 1 year from the north side of the tree and the other year the other side of the tree. Now, collection of leaves, when we are collecting medicinal plants leaves, the mature leaves should be collected before the flowering, not after the flowering of a medicinal plant. The source plant should not be ripped off the leaves completely. Certain percentage of leaves should be left to ensure normal physiological process. We can see in the pictures, like for the leaves, what to harvest. The fresh leaves, juvenile leaves should be harvested, and the disease and damaged leaves should be not taken off for the collection purposes. And now the points come how to harvest leaf, how we have to, you can see the first feature the collector, it is collecting पूरा का पूरा उसने branch को disturb कर लिया, तो we should not collect this purposes. हमको एक single leaf को तोड़ना है proper तरीके से to harvest the leaf. This comes under the good agriculture practices while collecting the medicinal plants. Now here we can see how to store the planting material. We have collected bark, we have collected the underground part, we have collected the leaves. Now the things comes how to process and how to store the material. So make the drug more potent or diminish its toxicity, eliminate unwanted properties, improve the smell and taste of the drug, and assure better storage and shelf life. Because whatever planting material you have collected, 10-15 दिन में आपने एक प्लांटिंग मटेरियल कलेक्ट किया है, अगर उसका बेटर स्टोरेज नहीं होगा, शेल्फ लाइफ नहीं होगी, तो इट इज ऑफ नो यूज, क्योंकि ह्यूमिडिटी उसमें नहीं होनी चाहिए. तो दीज आर सम ऑफ द स्टोरेज गाइडलाइंस फॉर मॉइस्चर. द एक्सेसिव मॉइस्चर कॉजिंग फंजाई, मोल्ड्स एंड पेस्ट टू डेवलप इन द हर्ब्स. टू कंट्रोल द रिस्क, मॉइस्चर मस्ट बी एलिमिनेटेड. तो मेडिसिनल प्लांट्स को जो 15% मॉइस्चर कंटेंट इज परमिसिबल इन केस ऑफ रूट्स. Airtight packaging is desirable. इसके लिए हम wooden boxes use कर सकते हैं. Drums and earthenware jars being used. इतना dry material हमारा होना चाहिए, moisture content उसमें नहीं होना चाहिए. Storage guidelines for temperature. All the raw herbal drug must be stored at cool place between 18 to 25 degrees centigrade. Shelf life के लिए prolonged shelf storage is labor consuming and may lead to the deterioration in quality. ये हम लोगों ने स्टोरेज किया है. तो फैक्टर्स अफेक्टिंग द क्वालिटी ऑफ हर्बल प्रोडक्ट, व्हाट आर द कंजर्वेशन मेजर्स, प्रोटेक्शन अगेंस्ट लाइट्स, लो टेम्परेचर, लो ह्यूमिडिटी, सुटेबल पैकेजिंग टाइप एंड करेक्ट टैग टू बी यूज्ड. बिकॉज हम लोगों ने टैगिंग नहीं सही की, तो एक प्लांटिंग मटेरियल कैन बी मिक्स विद द other planting material. These are the way how the medicinal plants can be stored in a rack or in a airtight glass jars. Now the quality consideration, when we coming to the botanical identity must be established before a plant species is collected from the wild. So this is one of the most important. And now the species collected should be same as described in various authentic reference books like of healthy plants, healthy individual of desired plant species should be harvested, and plants which are infested with insects, pests, bacteria, viruses should be avoided. Now the weather condition, when we go for a collection, what should be the weather condition for the collection purpose? Harvesting should not be done during rain because इसमें moisture content उसमें high होगा and this will encourage fungal attack. Wet plant material may be dried in intermittent light and shade. Collection should not be done during early hours to avoid dew. Right place for collection. Collection of medicinal plant produce should be done only from places which are clean and free from any possible exposure to insects, chemical, toxic gases, sewers, automobiles, etc. और हम लोगों को ये भी ध्यान रखना है when we should not harvest the plants from close to roadsides because of exposure to vehicular exhaust might, because nearby जो road या highways के nearby से हम लोग if we go for the collection, so there will be a high carbon dioxide content and this will be unsuitable for human consumption. When we are collecting the medicinal plant, the habitat management, हम लोगों को collection करना है ये लेके नहीं कि आज हमने collection कर लिया और उस area को पूरा destruct कर लिया. Damage to habitat of the species is to be minimized. It should be minimum. Climbers should be harvested in a way so as to cause least disturbances to the associated plant species. These are the sustainable use. Here the picture depicts, do not collect all good looking fruits, but leave some on the ground for regeneration. क्योंकि उससे fruit जमीन पे गिरेगा, उससे फिर जो seed होगा तो natural regeneration हो सकता है. Only collect fruit from trees and leaves and other. हमको क्या करना है कि सारे फ्रूट्स एक साथ नहीं कलेक्ट करने, कुछ ट्रीज को हम लोगों को छोड़ देना है, कुछ फ्रूट्स हम लोगों को ट्रीज में ही छोड़ देना है. We also need to prioritize a species suitable for cultivation in specific areas in consultation with local expert. Prepare a database, establish network of medicinal and ethnobotanical garden with nursery and research facility, strengthen state medicinal plant board and enhance the scope and coverage. Now we have talked about the good agriculture practices. What are the good agriculture practices? How we have to collect the material. Now coming up, first I will take up some medicinal plant of Himalayan region which are need for conservation, and then we will talk about the region where we are sitting at today, the tropical region. So some of the important medicinal plant of Himalayan region under conservation. This is one herb which is found in very high altitude in the hills of Uttarakhand and Himachal Pradesh. This is known by the name of Gentiana kuru, and it is also known as Ramban. It is used for the treatment of digestive disorders. The main reason for focusing on this plant is that there was one news like 61 साल बाद उगा Gentiana, फिर से हम लोगों को एक इसका natural regeneration मिला. तो इसी तरह से कई species हैं जो which were used to in our biodiversity, but slowly coming into the category of rare, endangered and threatened plants. Similarly, the Hippopea salicifolia, the seabuck thorn, it has three kinds of three species. So the whole area of Uttarakhand, you have seen, like there is a concept of a highway, a two-way highway, four-way highway. Now there is a six-way highway, and for this in Himalayan region, what we are doing, we are destructing the whole natural vegetation. Previously, when for the people going for the Char Dham Yatra, the Badrinath place, the whole side, the left and right side of the highway was occupied with this species, which is very rare, and now the whole habitat of this Hippopea salicifolia has been destructed. The medicinal use of commonly it is known as Hippopea salicifolia, commonly known as seabuck thorn. It was used for skin diseases. It was for anti-aging and regeneration of scale sets. It was so important species, but we have destructed the whole habitat of this medicinal plant. In China, this there are so many products which are manufactured with this sea buckthorn. They are exporting the thousands of varieties of products, excluding medicinal plant. This species is so famous in China, Russia, and Mongolia, and they are using it, producing it, and consuming it. But we in India, we are having the natural biodiversity of the species, and we are destructing the natural habitat, and slowly the species is getting into the endangered and extinct category. So if China can use so much product from this species, we, the Himalayan region, we are having rich biodiversity of this species. Why the local people can't have their livelihood generation from these kinds of products? Now, the recently, I think the 10 days before only, I just got one video in my WhatsApp about the rhododendron that are very useful species in the protection of COVID-19. हम लोग अभी तक काल में गिलोय वगैरह में ही थे कि ये काढ़ा पिए, ये आयुर्वेदिक करें, हल्दी का दूध पिए ये. तो अभी ये रिसेंटली आया है कि rhododendron, it is the state flower of उत्तराखंड, हिमाचल, अरुणाचल प्रदेश, सब जगह होता है. तो इसका जो जूस है, that is useful for COVID-19 treatment. These are the different species of rhododendron which are found in upper hills of India. Now the Texas baccata, Taxol, हम लोगों ने, सभी लोगों ने सुना होगा. It is an evergreen tree and it is also a medicinal purpose whose biodiversity is threatened in the Himalayan region. Now, some important medicinal plants of tropical region conservation. Then coming again to the COVID-19, हम लोगों ने देखा होगा कि जब COVID-19 था तो 90 रुपए की एक कीवी मिल रही थी. कह रहे थे कि हमें विटामिन सी चाहिए तो कीवी खाएं. डॉक्टर्स ने हम लोगों को कहा है कीवी खाने के लिए. तो we in India having so many other, हम लोग substitute नहीं ढूंढते. Like in case of Roha carambola, the fruit are very rich source of vitamin C, और इसमें citrus fruit के हिसाब से कई higher content होता है vitamin C का. तो ये, this was the species, this species, the Roha carambola, its fruits and flowers 4 times in a year. अगर आप चाहें तो ये हर season में available हो सकता है, but most commonly हमको market में monsoon season में दिखता है. When we were small, we used to see this species in front of the village girls' college. The girls used to eat it with the salt and consume it. But now the today generation, they will not able to identify these kinds of species. अगर हम आज की generation से पूछेंगे तो the child will be knowing kiwi क्या होता है, cherry क्या होता है, strawberry क्या होता है, but carambola क्या होता है, nobody will identify. अब identification नहीं होगा तो cultivation नहीं होगा, cultivation नहीं होगा तो species will, the biodiversity of this species of this important medicinal plant will be in danger. So we should, being from this botany background, being from forestry background, we should have this internal feeling to conserve the biodiversity of that species which was prevalent during our grandparents time. क्योंकि आप लोगों ने देखा होगा कि अब तो सीमेंटिंग पर्पस हो गया गांव में भी. प्रीवियसली जो हम लोगों का बरंडा होता था नानी दादी का, उनके किचन गार्डन में ऐसे प्लांट्स होते थे. आटोकार्पस लकूचा हो गया या कॉर्डिया मिक्सा हो गया, उसका अचार बना रहे हैं, पिकल्स बना रहे हैं, करौंदा है. तो these are the species which are also a rich source of vitamin C, but now slowly and slowly losing their biodiversity. This is the Cordia mixa. It beneficial effect in the cough, in chest disease, ulcer, lung disease, constipation and stomach ache. This species की मैं बात कर रही थी कि हम लोगों के यहाँ गांव में जब जाते थे तो किसी के बरंडे में this species used to be planted over there. और इसका, this is very famous for making a pickle. And there is one story which I recently heard. Still the villagers, rural people use the gum of this product. इसमें एक गम होता है इस कॉर्डिया मिक्सा में. Still the rural people, जो boys वगैरह गांव में रहते हैं, काइट वगैरह जुड़ाने के लिए कॉर्डिया मिक्सा का गम यूज करते हैं. And it is also very important medicinal species. Even I heard the Akbar Killa in संगम एरिया में जो अकबर किला है, एंड व्हेन द हम लोगों को पता ही है कि सीमेंट तो अब आया है, रिसेंट कंस्ट्रक्शन उसमें है, तो people used to join the material with the glue of this Cordia mixa. The third species is Spondeus spinnata, useful for dysentery, stomach ache, fever, ringworm, earache में इसका भी सॉरी कॉर्डिया मिक्सा को नहीं, स्पॉन्डियस स्पिनाटा को वाइल्ड मैंगो कहते हैं। और इसका ट्री भी हम लोगों के गांव एरिया में जब जाते थे तो बाहर बरंडे वगैरह में लगाया रहता था। और इसका वाइल्ड मैंगो इसलिए कहते हैं क्योंकि इसका शेप छोटे छोटे मैंगो से मिलता है और इसका पिकल्स बहुत टेस्टी पिकल बना करता था। पर अब ये बहुत कम ये जो मेडिसिनल प्लांट है जो डाइसेंट्री डायरिया वगैरह में यूज होता था। दीज आर द ट्रीज नॉट, हम लोग जब भी मेडिसिनल प्लांट का हम लोगों का कॉन्सेप्ट आ जाता है तो हम लोग वही देखते हैं, सर्वगंधा हो गया, कालमेघ हो गया, छोटे छोटे हर्ब्स हमारे दिमाग में आता है। पर जैसे मैंने फर्स्ट स्लाइड में ही कहा था, इवन 99% प्लांट्स आर मेडिसिनली यूज। हम लोगों को बस एक्सप्लोर करना है कि इनमें क्या मेडिसिनल यूज है। Now this Artocarpus lacucha antibacterial and antioxidant, आप लोगों ने देखा होगा ये फाइकस उस पे आता है, मोरेसी फैमिली का प्लांट है। पहले गांव के जो चबूतरे होते थे और गांव के जो स्कूल्स होते थे उनके बाहर आर्टोकार्पस लकुचा वगैरह का पेड़ लगा रहता था। बट अब जैसे सीमेंटिंग का कॉन्सेप्ट आ गया है तो अब जब इसका फ्रूट्स गिरता है बरसात में जुलाई के मौसम में तो बहुत गंदा कर देता है बरंडा वगैरह, तो इसलिए कोई इसको लगाना नहीं चाहता है, अब लगाते नहीं है। और इसकी जो सीड वायबिलिटी होती है ये सिर्फ होती है 1 हफ्ते की। अगर इस प्लांट का जैसे क्या होता था, नेचुरल रीजनरेशन होता था, जब सीड गिरता था ट्री से नीचे गिरता था तो नेचुरल रीजनरेशन हो जाता था और प्लांट जर्मिनेट हो जाते थे। अब क्या होता है कि पूरा सीमेंटिंग पर्पस है या गांव में गिर भी रहा है तो इसीलिए इसकी जो पॉपुलेशन है वो बहुत कम देखने को मिलती है और स्लोली स्लोली बायोडाइवर्सिटी इज इफेक्टिंग। This is the one species Stereospermum sovalensis, stomach ache, malaria, liver, asthma, इसमें nervous diseases वगैरह में यूज होता है। One of the important ingredient of Dasmodarish। इस प्लांट का जो आप ये देख रहे हैं जो कैप्सूल है, इसके लोग वहाँ फैजाबाद एरिया में मैंने आपको बताया कि वहाँ के लोग इसे माइग्रेन के लिए यूज करते हैं। एंड इवन द प्रेग्नेंट लेडीज या जो यूजली विथ फीमेल्स जिनके कमर वगैरह में दर्द होता है, इसकी जो ये लड़ी है इसको एक मोती की तरह पिरो दे देते हैं और कमर में पहनती है तो दे हैव वन मिथ कहिए या उन लोगों को सच में आराम मिलता है कि इस स्पीशीज को जब कमर में लगाते हैं तो पेन रिलीविंग है। माइग्रेन में सिर में ऐसे लगा देते हैं इसकी लड़ी को तो ये ठीक हो जाता है। तो यही हम लोग जैसे इंडियन ट्रेडिशनल सिस्टम ऑफ मेडिसिन की बात करते हैं तो हमारे पास इतने दादी नानी के नुस्खे हैं कि हम लोग वी आर सो रिच इन दिस स्टोरीज। नाउ द वन अभी हमने सारी ट्रीज की बात की। एक नाउ कमिंग टू द स्मॉल प्लांट फाइलेंथस एमेरस, ये हिंदू आयुर्वेदिक सिस्टम ऑफ मेडिसिन में यूज होता था एंड डायबिटीज लीवर वगैरह में होता है। आपने बरसात में देखा होगा वो छोटा छोटा सा एक प्लांट हर्ब इतना छोटा सा होता है और बहुत कॉमन होता है। डेग्रेडेड लैंड जो होती है उस एरिया में अगर हम इसे लगाएं तो एरिया कैन बी रिक्लेम। तो इस तरह की मेडिसिनल स्पीशीज हम लोग कुछ ऐसी स्पीशीज डिस्कवर करें जो यूजेस है, जो जिनके बहुत यूजेस है और उनकी बायोडाइवर्सिटी हमको प्रोटेक्ट करनी है। तो different formulation of plant parts are prepared and used by rural man to cure a particular diseases. Now there are some diseases like fever, headache hai, and what are the plants which we can use and what parts we can use. For example, in case of Cassia tora, the part uses seed और ये fever के उसमें use होता है. Similarly for headache, Santalum album paste is used. वाइटेक्स निगेंड और निर्गुंडी है, इसका लोग हेज की तरह भी जैसे बायोफेंसिंग के लिए भी यूज करते हैं. लीव्स इज यूज फॉर हेडेक, इवन एसेंशियल ऑयल भी इससे निकलता है. नाउ फॉर लीवर डिसऑर्डर्स, अभी जो मैंने आपको फाइलेंथस एमेरस दिखाया फॉर लीवर डिसऑर्डर, इट्स लीव्स आर यूज और एंड्रोकाल्मी का लीव्स आर यूज. कोल्ड कफ के लिए जो हम लोग फ्रूट्स यूज करते हैं, तो ये सब हम लोग देख सकते। अस्थमा है, स्नेक बाइट है, तो ये सब प्लांट यूज कर रहे हैं अस्थमा के लिए। करक्यूमा लॉन्गा है, एलेंथस एक्सेल्सा है, स्नेक बाइट वगैरह के लिए बुचेनिया लंजन है, चिरौंजी है। जो चिरौंजी स्टिल अगेन द जर्मिनेशन ऑफ दिस स्पीशीज इज वेरी पुअर। और अब आप लोग देखेंगे कि कहीं भी आपको चिरौंजी का ट्री देखने को नहीं मिलेगा, बिकॉज जब इसके जर्मिनेशन में ही प्रॉब्लम है, स्लोली वी आर लूजिंग दिस इम्पोर्टेंट medicinal plant whose stem and bark is of great importance. Now we can say there are two medicinal plant की बात कर ली हमने, कह दिया endanger में जा रहे हैं, extinct में जा रहे हैं। Now the questions come, how we can conserve this species, how we can conserve by in situ conservation, by research and development projects, by marketing value addition, information and education. The most important is in situ conservation and ex situ conservation. Now the technical term is in situ conservation, means conservation of ecosystem and natural habitat, and the maintenance and recovery of viable population of a species in their natural surroundings. Natural surrounding means कोई बायोस्फीयर रिजर्व है, कोई नेशनल पार्क है, तो वहां हम लोग कोई डिस्टरबेंस नहीं कर रहे हैं। नेचुरल सराउंडिंग्स में पॉपुलेशन नेचुरल रीजनरेशन हो रहा है, प्लांट्स अपना खुद प्रोटेक्ट हो रहा है, हम लोगों को कुछ नहीं करना है। नेचुरल रीजनरेशन में इन सिटू कंजर्वेशन हो रहा है। नाउ नेक्स्ट टेक्निकल टर्म इज एक्स सिटू कंजर्वेशन। एक्स सिटू कंजर्वेशन मींस ऑफ साइट कंजर्वेशन, कंजर्वेशन ऑफ ऑल लेवल्स ऑफ बायोलॉजिकल डाइवर्सिटी आउटसाइड देयर नेचुरल हैबिटेट थ्रू हम लोग बोटैनिकल गार्डन एस्टैब्लिश कर सकते हैं, जर्म प्लास गार्डन एस्टैब्लिश कर सकते हैं, अपनी नर्सरी बना सकते हैं। तो दिस इज एक्स सिटू कंजर्वेशन जो हम लोग करते हैं। In situ conservation, it is sustainable harvest of selected species recovery for rest लिए, and ex situ conservation germ plas bank बना दिया या आपने medicinal plant garden बना दिया, तो आप क्या off site conservation कर रहे हैं या development of propagation procedures। और जब हम ex situ conservation की बात करते हैं तो it is conservation through cultivation, planting से करेंगे या seed से करेंगे। Supply of planting material to farmers, if some farmers is interested to establish some medicinal plant cultivation in his own field, so supplying the planting material, good planting material to that farmers, and giving training and technical assistance in medicinal plant cultivation, and socio-economic evaluation before and after adoption of commercial cultivation by farming and village communities। ये एक स्लाइड थी, यही दिखाया कि हाउ वी प्रोपोगेट द प्लांटिंग मटेरियल, कटिंग से करते हैं या सीड से करते हैं। जब हम लोग बीज से करते हैं तो उसको प्रोजेनी कहते हैं, और जब कटिंग से करते हैं तो रैमिड्स करते हैं। दिस इज आई थिंक नॉट वेरी यूजफुल फॉर यू पीपल। नाउ इंपॉर्टेंट टूल्स, जब हम लोग मेडिसिनल प्लांट कल्टीवेशन करते हैं तो कलेक्ट सीड्स ऑफ मेडिसिनल स्पीशीज फ्रॉम हेल्दी स्ट्रेट ट्रीज एंड गुड क्लीन बोल। हम लोग एक कॉन्सेप्ट लाते हैं प्लस ट्री का, किस ट्री से हम लोगों को मेडिसिनल प्लांट कलेक्ट करना है। हेल्दी होना चाहिए, स्ट्रेट होना चाहिए, क्लीन बोल होना चाहिए, डिजीज डैमेज नहीं होना चाहिए। जब हम लोग मेडिसिनल प्लांट को जर्मिनेट करने के लिए जाते हैं तो मेक होल्स इन पॉलिथीन बैग बिफोर फिलिंग सॉइल मिक्सचर टू ड्रेन एक्सेस वाटर। कई बार क्या होता है कि पॉलिथीन बैग जो हमें मार्केट में मिलती है उसमें होल्स नहीं होते हैं, तो हम लोगों को उस पॉलिथीन में होल्स कर देने हैं वाटर ड्रेनेज के लिए। जब भी हम लोग मेडिसिनल प्लांट को हमने बेड में जर्मिनेट कर दिया तो डू ट्रांसप्लांटिंग इन द स्टेज। व्हाट इज ट्रांसप्लांटिंग? मींस ट्रांसफर ऑफ प्लांट फ्रॉम बेड टू पॉलीबैग और फ्रॉम पॉलीबैग टू बेड और फ्रॉम वन प्लेस टू अनदर। ट्रांसप्लांटिंग मीन जैसे ट्रांसफर करते हैं ना, तो हमने एक जगह से दूसरी जगह जब भी हम मेडिसिनल प्लांट को करें तो इट शुड ऑलवेज इन शेड एंड मॉर्निंग के टाइम पे करें या इवनिंग के टाइम पे करें। बहुत ज्यादा जब सनरेज होती है दिन के टाइम में ना करें। एंड प्लांट सीडलिंग विथ हाई रूट शूट रेशियो। जब हम लोग इसकी, जैसे हम लोगों ने बेड में जर्मिनेट कर दिया, अब हमको इस प्लांट को कहीं पॉलिथीन में लगाना है या कहीं और लगाना है, तो प्लांट सीडलिंग विथ हाई रूट शूट रेशियो, एक बराबर का रूट शूट रेशियो होना चाहिए, बहुत छोटा सा प्लांट नहीं होना चाहिए। Take control measure against pest disease as soon as infestation begins. पॉलिथीन बैग की बात कर रहे थे तो इन नर्सरी वी यूज दीज काइंड्स ऑफ पॉलिथीन। आप देखेंगे कि छोटा मेडिसिनल प्लांट है तो छोटी पॉलिथीन यूज करेंगे, एंड if the plant medicinal plant जैसे कई trees are having medicinal value, so we are using a polythene bag of this kind of size. Then after important do's, there are important don'ts. हम लोगों को क्या नहीं करना है जब मेडिसिनल प्लांट नर्सरी एस्टैब्लिश कर रहे हैं? डोंट एस्टैब्लिश नर्सरी वेयर इरिगेशन वाटर इज इनएडिक्वेट और अनसर्टेन। जब भी हम लोग कोई नर्सरी बना रहे हैं या अपना किचन गार्डन कर रहे हैं जो भी, तो हम लोगों को एक एश्योर, वी शुड हैव एश्योर वाटर सप्लाई सिस्टम फॉर इरिगेशन। डोंट कलेक्ट सीड ड्रॉप्ड ऑन द ग्राउंड। जैसे हम लोग मेडिसिनल प्लांट के लिए वो कोई भी सीड ऐसी हम ना कलेक्ट करें जो ट्री से गिर चुकी है, क्योंकि ट्री से गिर चुकी है तो या तो ओवर मैच्योर फ्रूट हो गया, या हो सकता है वो डिजीज डैमेज हो, उसमें कीड़ा लगा हो या फंगल अटैक हो। तो हम लोगों को मेडिसिनल प्लांट के लिए जो नीचे गिरी ड्रॉप्ड सीड्स है, वी डोंट हैव टू कलेक्ट दैट। डोंट सो सीड टू डीप और टू शैलो। बहुत ज्यादा ऊपर भी नहीं सीड को बो करना। एक जो कॉन्सेप्ट होता है कि जो सीड होती है उसके 1 और 2 हमको उसको ग्राउंड लेवल पे नीचे जर्मिनेट करना होता है। बहुत ज्यादा ऊपर भी नहीं रखना है, बहुत ज्यादा डीप भी नहीं शो करना है, बहुत ऊपर भी नहीं शो करना है उसको। Don't keep transplant in the sun, keep them in shade for 2 weeks till they establish. Don't plant trees that were harmful insect, birds and near schools and house, etc. The polythene bag में we germinate the medicinal plant, and another we germinate in fruit trainers, and there are different shapes of fruit trainers. इनको जो है इनको हम cells कहते हैं, कोई 64 cells का है, कोई 25 cells का है। तो these are the types of root trainers where we can germinate the medicinal plants. What are root trainers? Root trainers are conical tube made up of plastic. The inner walls of the root trainer have a vertical grooves और ridges होते हैं, जो आप लोग ये देख रहे होंगे। इसमें जो vertical ridges होता है ये root coiling को prevent करता है, क्योंकि कुछ मेडिसिनल ट्रीज ऐसे होते हैं जिनको हम लोग जैसे जर्मिनेट कर रहे हैं तो कुछ टाइम बाद जब उनकी सीड्स होती है तो पॉलिथीन बैग में क्वाइल हो जाती है। रूट क्वाइलिंग की प्रॉब्लम होती है। जब उनको फील्ड में लगाते हैं तो रूट क्वाइलिंग से प्लांट डैमेज हो जाता है जब उनसे पॉलिथीन बैग से निकालते हैं। तो इस पर्पस के लिए रूट ट्रेनर्स होते हैं जिनमें वर्टिकल रिजेस ग्रोव्स होते हैं और रूट क्वाइलिंग प्रिवेंट करते हैं। शेड हाउस की बात करें तो शेड हाउस इज यूज्ड फॉर हार्डनिंग ऑफ फ्रेशली प्रोपोगेटेड प्लांट्स। ये एग्रोनेट्स होते हैं जिनकी परसेंटेज 30, 75, 90% होती है। अलग अलग उससे परसेंट शेड दे प्रोवाइड। ये आप लोग देखेंगे डिफरेंट शेड्स एंड साइजेस ऑफ मिस्ट चैम्बर्स। ये मिस्टिंग के लिए देखेंगे आप, एक एग्जॉस्ट फैन है, फिर यह मिस्टिंग है। इनमें टाइमर लगा होता है कि अगर हर 15 मिनट, गर्मियों में हर 15-20 मिनट बाद ऑटोमेटिकली द मिस्टिंग ऑफ मेडिसिनल प्लांट्स के ऊपर मिस्टिंग शुरू हो जाती है। तो दीज आर now the another important concept of medicinal plant which by my observation in the Allahabad and Kaushambi region, the medicinal plant cultivation under tree orchard. This concept which came to our mind was in we have seen in Allahabad area कि यहाँ इतने सारे महुआ के गार्डन ऑर्चार्ड हैं, और कौशाम्बी में चले जाएं तो गुआवा के ऑर्चार्ड है, और उनके अगर आप हाई ऑल्टीट्यूड में चले जाएं एप्पल ऑर्चार्ड हो जाते हैं। साउथ इंडिया में चले जाएं कोकोनट ऑर्चार्ड है। तो इसमें क्या हम लोगों ने देखा कि जो नीचे की स्पेस रहती है वो वेकेंट रहती है। जैसे आप ये देखिए कि इतनी ज्यादा स्पेसिंग दी हुई है, इसमें 4x4 की है, 3x3 की है। तो इसके नीचे की एरिया जो सारे वेकेंट होते थे, गुआवा हो गया, महुआ जो ईस्टर्न यूपी का इंपॉर्टेंट मेडिसिनल प्लांट है, तो मैंगो ऑर्चार्ड हो गया। तो दीज स्पेसेस वेर वेकेंट। तो इनके नीचे क्यों ना मेडिसिनल प्लांट कल्टीवेशन किया जाए, क्योंकि देयर आर सो मेनी मेडिसिनल प्लांट्स व्हिच आर शेड लविंग। तो उन मेडिसिनल प्लांट को अगर इनके नीचे हम लोग लगाते हैं तो जो फार्मर है वो एक्स्ट्रा इनकम जनरेट कर सकता है। तो एक कॉन्सेप्ट हम लोगों ने यहाँ पे, तो ये देखिए ये बैंगन वगैरह लगाया हुआ, मीन्स वेजिटेबल भी उगा सकते हैं एज वेल एज मेडिसिनल प्लांट के नीचे कल्टीवेट कर सकते हैं। तो एक हम लोगों ने ये एक प्रोजेक्ट किया था मेडिसिनल प्लांट कल्टीवेशन अंडर ट्रीज। इसमें महुआ गुआवा जो इधर के ऑर्चार्ड है कौशाम्बी एंड इलाहाबाद के, उसमें एक ये स्टडी की गई थी। तो दिस इज वन न्यू कॉन्सेप्ट व्हिच आई जस्ट वांटेड टू हाईलाइट हियर। इफ somebody is having this orchard, they can utilize the space. नाउ थिंग आता है कि इसके नीचे लगा क्या सकते हैं, तो जो शेड लविंग है जैसे एंड्रोग्राफिस पेनिकुलेटा है, जिंजर है, करक्यूमा, हल्दी, अदरक वगैरह, ये सब हम लोग सर्पगंधा वगैरह, एस्पैरागस रेसिमोसस, ये सतावर वगैरह हम लोग इसके नीचे लगा सकते हैं। इंपॉर्टेंट मेडिसिनल प्लांट इन डिमांड इन उत्तर प्रदेश आंवला है, अरहर, बहेला, गिलोय, शंखपुष्पी, ये सब दीज आर द इम्पोर्टेंट मेडिसिनल प्लांट्स। अब बहुत ज्यादा मैं नहीं दिखाऊंगी, जैसे एकोरस कैलमस है, वच है, तो ये इसका एक इकोनॉमिक्स रखी है क्या है। If somebody is interested, I can share my presentation with that fellow, क्योंकि इकोनॉमिक्स का actually when we go with जब हम लोग किसान वगैरह को एक लेक्चर देते हैं तो दे आर मेनली इंटरेस्टेड कि लगाएंगे तो कितना इनकम हम लोगों को मिलेगा। तो दिस स्पीशीज, ये जैसे अश्वगंधा है, लो वाटर रिक्वायरमेंट है, सैंडी लूम, लाइट सॉइल वगैरह है, तो दीज आर द स्पीशीज जो इसका एक प्रोपोगेशन टेक्निक लिखी है कि व्हाट काइंड ऑफ वेदर कंडीशन एंड हाउ मच सीड सोइंग इज रिक्वायर्ड, व्हाट काइंड ऑफ इरिगेशन एंड व्हेन इज द फ्रूटिंग एंड फ्लावरिंग ऑफ दीज स्पीशीज। कालमेघ एक इम्पॉर्टेंट है, तो फाइन फील्ड प्रिपरेशन चाहिए, अप्रोप्रिएट ड्रेनेज चाहिए, हार्डी प्लांट है, गो ऑन वेराइटी ऑफ साइल, these are the, ये हल्दी के लिए है, शेड लविंग है। तो अगर if some of you students are having some orchard in your villages, तो you can go for the haldi cultivation under that orchard। intercropping with trees ये आराम से हो जाती है। एलोवेरा है, इसका भी कोई अगर if somebody is इंटरेस्टेड फॉर मेडिसिनल प्लांट बिजनेस, तो एलोवेरा बहुत ही इजीली ग्रो कर सकते हैं। और कई जैसे डाबर है, कई एजेंसीज हैं जो इसका प्लांटिंग मटेरियल खरीदने को तैयार रहते हैं, बिकॉज आजकल आप देखेंगे अंडर आई जेल है या हेयर में यूज कर रहे हैं लोग, फेशियल क्रीम्स वगैरह में, तो बहुत ज्यादा इन यूज है एलोवेरा। अब ना ट्रेडर्स इन्वॉल्व इन सेल ऑफ मेडिसिनल प्लांट, तो लखनऊ में आप देखेंगे दीज आर द ट्रेडर्स। अगर आप एलोवेरा या सतावर वगैरह जैसे कोई किसान या कोई स्टेक होल्डर ग्रो कर लेता है, तो वही फर्स्ट क्वेश्चन दे आस्क इज वेयर वी हैव टू सेल। अदर पार्टनर्स इन मेडिसिनल वर्क आर स्टेट मेडिसिनल प्लांट बोर्ड, नेशनल मेडिसिनल प्लांट बोर्ड एंड द विलेज फॉरेस्ट मैनेजमेंट कमेटी है, स्टेट फॉरेस्ट डिपार्टमेंट है, मिनिस्ट्री ऑफ एनवायरमेंट, फॉरेस्ट एंड क्लाइमेट चेंज है। एक ये एनएमपीबी का इनिशिएटिव है फॉर मेडिसिनल प्लांट ई चरक। दिस इज ऑनलाइन पोर्टल कम एंड्रॉइड बेस्ड एप्लीकेशन फॉर ट्रेडिंग ऑफ मेडिसिनल प्लांट। इसमें आप देखेंगे कि ये कितने नंबर ऑफ यूजर्स हैं, रजिस्टर्ड ई चरक में कितने नंबर्स हैं। तो इसमें आप लोग मेडिसिनल प्लांट अगर कोई बेचना चाह रहा है तो ये वाला ऐप अप्लाई यूज कर सकता है। इट इज अवेलेबल इन 7 languages। The other important addresses of the funding agencies who deals with medicinal plants, जैसे national medicinal plants में some student is interested for submitting the project, so they can go for funding from national medicinal plant boards and others agencies. So I think and good agriculture practices. I thought of not showing the cultivation of this common species. I have just dealt my subject dealing with some tropical as well as some temperate, some species whose biodiversity is endangered, and we as a human, we should save these medicinal plant species for our future generation. So thank you so much for giving me this opportunity. I once again wants to thank the organizers of SS Khanna Government Degree College for organizing this event and connecting us with so many people, and thank you so much.
Thank you, ma'am. Thank you for such an informative lecture. Thank you once again. We will go ahead and take some time for the questions. I would like to invite Mr. Shivam Mishra, Assistant Professor of Zoology of this college, to conduct this part of the session. Over to you, Shivam.
Okay, good afternoon, ma'am, and thank you for inviting me. So, thank you so much, ma'am, for your informative lecture. And dear participants, if you have any question, you can ask in chat box. Ma'am, first of all, one question is there. Ki, how we can choose which plant is medicinal? Yeah, which plant can be used as medicinal purpose? Am I audible, ma'am? Am I audible? Am I audible?
Anita ma'am, hello.
Please let me
Hmm?
Am I audible, ma'am?
Ma'am,
Yes, you are. Yes, you are, Shivam.
Okay.
Anita ma'am.
Anita ma'am, please unmute yourself. Anita ma'am, please unmute yourself. Ma'am is not responding right now.
Yes, please.
Okay, ma'am, one question is there, how we can identify which plant we use as medicinal?
See, for example, when I was talking about some of the medicinal plants of temperate species and some of the medicinal plants of tropical species, already it is in the literature which medicinal plant is used for which purpose and which part of a plant is used for which purpose. Like for example, in rural community, when we are going, we are getting so many examples from the rural people. For example, local people बात करते हैं कि हम लोग ये प्लांट इस पर्पस के लिए यूज कर सकते हैं. ये उन लोगों के अपना वो है, एक्सपीरियंस है कि जैसे बोन ब्रेकिंग के लिए आप लोग पहाड़ों में चले जाएंगे तो वहां जहां डॉक्टर्स वगैरह नहीं होते हैं तो वहां का हर आदमी वैद्य होता है. वो कहता है जी हमारे जो लोकल वुमेन है उसने ये नहीं देखा है कि जब उसकी डिलीवरी हुई है तो डॉक्टर्स के यहाँ ले गए तो अपने उन लोगों के जो रूरल थ्योरीज है वो उससे इलाज करते हैं. और बोन ब्रेकिंग का फॉर एग्जाम्पल वो लोग कई स्पीशीज होती है उसका लेप वगैरह लगाते हैं, किसी स्पीशीज का लीव्स कलेक्ट करके लेप लगा लेते हैं. तो इट इज नॉट वी हु आर डिसाइडिंग कि विच मेडिसिनल प्लांट इज यूज फॉर विच पर्पस. जैसे फॉर एग्जांपल अर्जुन का है तो इट इज वेरी मच इन द लिटरेचर कि इसका बार्क हम लोग यूज करते हैं, या सतावर आप लोग कर लीजिए, या गिलोय अभी जो कहा गया कोविड-19 आया तो सब लोगों ने कहा कि इसका काढ़ा पिएंगे तो इम्यूनिटी बढ़ेगी. तो दीज आर ऑल इन द लिटरेचर. हम लोग अगर महाभारत या रामायण वगैरह की टाइम करेंगे तो उस समय से ही हमारे वेदास वगैरह में कई तरह की वो दी हुई है कि विच मेडिसिनल प्लांट इज यूज फॉर विच पर्पस. और हम लोग जो है जैसे इसका एक टेक्निकल टर्म होता है बायो प्रोस्पेक्टिंग की वैल्यू एडिशन हम लोग कैसे उसका कर सकते हैं. जैसे हम लोग वाइटेक्स निगंडो की हम लोग बात करें, मैंने अभी एक स्लाइड में दिखाया था वाइटेक्स निगंडो leaves is used for this medicinal purpose. So we are using this species. जैसे हम लोग bio prospecting है, we are collecting the different extraction of this species and we are distillating the essential oil for this, and in later on studies we will identify how much quantity of essential oil was permitted from this different extractions.
Okay, thank you, ma'am.
मैम प्लीज अपना वीडियो ऑन कर लीजिए ना. एवरीवन रिक्वेस्टेड प्लीज वीडियो ऑन कर लीजिए फॉर फोटो क्लिक. शिवम तुम अपना कंटिन्यू करो क्वेश्चन, बस वीडियो ऑन.
ओके मैम, वन मोर क्वेश्चन.
अनीता मैम वीडियो ऑन कर लीजिए प्लीज.
इन सम एंटी एजिंग प्लांट, some plant which can be used as anti-aging.
Anti-aging,
yeah.
ये जैसे आंवला इज वेरी कॉमन. आंवला आप लोगों ने देखा होगा कि कहा जाता है कि मुरब्बा इसका खाएंगे तो एंटी एजिंग के लिए यूज हो सकता है। और जितने भी विटामिन सी वाले हैं, फॉर एग्जांपल कीवी हो गया या एब्रोहा कैरम्बोला हो गया या सिट्रस फ्रूट वगैरह खाएंगे तो दीज आर गुड फॉर एंटी एजिंग, विटामिन सी। मोस्ट कॉमन आप लोग जो जैसे एमली का ऑफिसिनालिस है, ये तो बहुत कॉमन है। इसको आप जैसे कैंडीज फॉर्म में यूज कर सकते हैं, मुरब्बा फॉर्म में यूज कर सकते हैं, इसका पिकल वगैरह यूज कर सकते हैं। तो इट इज वन ऑफ द एंड वेरी गुड एंटी एजिंग।
Okay. Can you please suggest some medicinal plant in Allahabad region?
यहाँ तो मैंने आपको एक स्लाइड दिखाई थी, गिलोय लगा लीजिए, अश्वगंधा लगा लीजिए, सतावर लगा लीजिए, और ये जैसे आप लोगों को अरगंधा, एंड्रोग्राफिस पैनिकुलेटा वगैरह ये सब तो है ही। और अगर ट्रीज वगैरह में लगाना है तो आप लोग जैसे इट्स डिपेंड कि आप लोग को फॉर विच पर्पस, फॉर किचन गार्डन पर्पस ही रिक्वायर्ड तो हम लोग कह देते हैं हल्दी अदरक लगा लीजिए आपके पीछे। अगर आपको मेडिसिनल यूज करना है तो सतावर कॉमन हो जाता है। आपको जैसे कई लोग होते हैं वो कहते हैं हम लोगों को भाई हैजिंग पर्पस के लिए करना है तो उसमें भी हम लोग सतावर यूज कर देते हैं। हाई ब्लड प्रेशर वगैरह के लिए ये यूज होता है। तुलसी है, और आंवला हो गया आपका, अर्जुन हो गया, ट्री ये आ जाता है। हम लोग फिर जैसे कुछ लेस कॉमन वगैरह में चले जाते हैं तो ये वाइटेक्स निगंडो हो जाता है आपका। तो इस तरह से है। तो इट्स डिपेंड कि आपका कॉन्सेप्ट क्या है, आपका किचन गार्डन कॉन्सेप्ट है या हैंगिंग गार्डन में है, विच पर्पस के लिए। और जैसे आई ऑलरेडी टोल्ड कि हम लोग एक ये मिथ है, कह देते हैं कि मेडिसिनल प्लांट हम लोगों ने डिस्कवर किया है कि नहीं किया है, वी कैन, हम लोग ये लेके चल सकते हैं 90% ऑफ द प्लांट्स forest are having some or other medicinal use. And ये डिपेंड करता है कि जैसे मटेरियल कोई लगाना चाहते हैं तो वी एज अ फॉरेस्ट रिसर्च सेंटर वी आर प्रोवाइडिंग प्लांटिंग मटेरियल ऑफ सम ऑफ द मेडिसिनल प्लांट्स व्हिच वी आर हैविंग। तो हम लोग ये प्लांट्स अपना जैसे प्रोपोगेट करते हैं और देखते हैं।
ओके, अगर कोई फार्मर जो है यूपी ईस्ट रीजन में अगर इसको एज अ बिजनेस पर्पस से ह्यूज अमाउंट में अगर कल्टीवेट करना चाहे तो कुछ आप सजेस्ट करना चाहेंगे कि यूपी इस रीजन में कौन सा मेडिसिनल प्लांट जो है उसकी प्रोडक्टिविटी ज्यादा बेहतर है और बिजनेस पर्पस से भी उसका आउटकम अच्छा निकलेगा?
देखिए जैसे अभी हम लोगों ने बताया यहाँ जो ईस्टर्न यूपी में चल रहा है, तो एक फूलपुर में जहाँ इफ्को के पास है हम लोगों का एक किसी किसान से टेकअप है, तो वो सतावर के पौधे का मतलब वो बनाता है, वैल्यू एडिशन करता है। और इसी को सतावर को आप लोगों ने देखा होगा, उसने अपना जो ये प्लांट है सतावर का, वो जो ये खादी वाले हैं उनसे टाई अप किया है। तो मतलब खादी वाले उनसे उनका जो वो कुछ वो बनाते हैं च्यवनप्राश टाइप ऑफ बनाते हैं सतावर का, तो उसको लेते हैं तो सतावर लगा सकते हैं। दूसरा एलोवेरा भी यहाँ का किसान लगा सकता है, एलोवेरा। और ये आप लोगों ने अभी देखा होगा जब ये एक कॉन्सेप्ट चल रहा था फॉरेस्ट डिपार्टमेंट का कि इतने लाख पौधे लगाने हैं जुलाई के सीजन में, जब हमारे माननीय मुख्यमंत्री जी ने एक स्पीशीज बहुत फोकस की थी, मोरिंगा ओलिफेरा, सैजन को। उन्होंने कहा था कि इसको सैजन को बहुत ज्यादा प्रमोट करना है। तो सैजन भी एक ऐसी स्पीशीज है जो यहाँ का ईस्टर्न यूपी का किसान लगा सकता है और इसका हर पार्ट यूज में होता है। इवन फ्रूट्स ईडेबल है और मेडिसिनल यूज भी है, और साउथ इंडिया में तो ये बहुत ही कॉमन है। तो इस स्पीशीज में मोरिंगा है, सतावर है, गिलोय है, एलोवेरा है, और इवन जैसे मैंने एक आपको कॉन्सेप्ट बताया फॉर कल्टीवेशन अंडर ऑर्चार्ड, हल्दी अदरक वगैरह, ये स्पीशीज करके भी एक द फार्मर्स कैन हैव सम इनकम जनरेशन का।
Okay, ma'am. Thank you. Participants, if you have any question, you can ask. Okay, एक क्वेश्चन है मैम कि व्हाट अबाउट आंवला, व्हाट अबाउट अगर इसको हम कल्टीवेट करना चाहें, बट एट दिस टाइम कि जो किसी भी फार्मर के लिए जो सबसे बड़ी प्रॉब्लम होती है, कल्टीवेट तो वो कर लेते हैं कहीं से भी वो नॉलेज लेकर के, तो आज के टाइम पे यूट्यूब पे बहुत सारी इंफॉर्मेशन है, बट जो नेक्स्ट प्रॉब्लम, मेजर प्रॉब्लम आती है इसके बाद क्या, कहाँ पर उनको सेल करना है, कैसे सेल करना है उनको, क्योंकि आत्मनिर्भर भारत के तहत बहुत सारे लोग अपना खुद का सेटअप करना चाह रहे हैं, जिनके पास लैंड है वो एग्रीकल्चरल फार्मिंग करना चाह रहे हैं। अगर वो मेडिसिनल प्लांट की करना चाह रहे हैं, करने के बाद वो एक्चुअली उनके पास जो मेन प्रॉब्लम आती है कि उसको सेल कहाँ करें, वो कैसे सेल करें वो।
मैंने आपको लास्ट स्लाइड्स दिखाई थी, ट्रेडर्स इन लखनऊ where the farmers, if they are cultivating the medicinal plant, they go for selling of this. फिर मैंने NMPB एक वेबसाइट आपको दिखाई, उसमें जा सकते हैं। Even the herbal industry like डाबर, इमामी, this खादी, ये आपको पहले से टेकअप करना पड़ेगा। किसान क्या करता है, वो फसल लगा देता है और फिर मार्केट ढूंढता है। तो किसान को ये करना चाहिए, पहले वो मार्केट ढूंढ ले, जैसे टाई अप कर ले इमामी, खादी या किसी से, या ई चरक वेबसाइट में अपना देख ले। इवन CMAP इंस्टीट्यूट इन लखनऊ, दे हैव द मार्केट फैसिलिटी, वहां जाके कांटेक्ट कर ले कि आई एम प्लानिंग टू प्रोपोगेट दिस हेक्टर्स और एकर्स ऑफ मेडिसिनल प्लांट, क्योंकि बहुत छोटे एरिया में लगा के होगा, मार्केटिंग होनी होगी। और फॉर, जैसे आपने बताया आंवला का, आंवला तो जैसे प्रतापगढ़, दे हैव डेवलप सो मेनी वेराइटीज वगैरह है, उनपे ये है। तो आंवला का तो जो लोकल वो होते हैं, जैसे SFDs, स्टेट फॉरेस्ट डिपार्टमेंट, या वैल्यू एडिशन वगैरह, लोकल विलेजेस अपने ही वहां एक इंडस्ट्री डेवलप करके, जैसे मैंने बताया सतावर का च्यवनप्राश बना रहे हैं। तो जो फूड प्रोसेसिंग इंडस्ट्री है, आंवला का मुरब्बा, कैंडी वगैरह बना ही रही है। प्लस इन प्रतापगढ़ एरिया आंवला का इतना ज्यादा स्कोप है कि दे कैन कॉन्टेक्ट there if somebody is interested for cultivating this species, और आंवला को भी they can have this agroforestry concept with आंवला, क्योंकि हम लोगों को बात करनी है इनकम जनरेशन की। आंवला लग रहा है, उसके नीचे और कोई शेड लविंग या कोई और मेडिसिनल प्लांट भी कर सकता है कि उसको एक फसल के साथ साथ दूसरे से भी कुछ इनकम जनरेशन हो सके। तो कॉन्सेप्ट शुड बी कि जैसे एग्रीकल्चर के साथ साथ मेडिसिनल प्लांट करे तो उसको दो उससे इनकम मिल जाएगी, एग्रीकल्चर क्रॉप से भी, मेडिसिनल प्लांट से भी। या एग्रो फॉरेस्ट्री कर रहा है तो फॉरेस्ट्री स्पीशीज है, फास्ट ग्रोइंग ट्री स्पीशीज लगा सकता है, फॉर एग्जांपल पॉपुलर यूकलिप्टस, और उसके अंदर एग्रो फॉरेस्ट्री कर सकता है, एग्रीकल्चर कर ले या मेडिसिनल प्लांट कल्टीवेशन कर ले। तो फिर एक क्वेश्चन और आ जाता है कि मैं इन ट्रीज के साथ अगर क्रॉप लगाऊंगा तो मेरा जो ट्री स्पीशीज है या जो मेरा एग्रीकल्चर है उसका थोड़ा बहुत जो वो है सेल है कम हो जाएगी, ग्रोथ कम हो जाएगी। तो वो थोड़ा बहुत होता है। फॉर एग्जांपल इन केस ऑफ यूकलिप्टस है, यूकलिप्टस के नीचे जो भी लगा रहा है तो जो ओवरऑल रिटर्न उसको 7-8 साल बाद मिलेगा यूकलिप्टस से। तो एक प्रॉफिट तो उसे यूकलिप्टस बेचने से हो जाएगा या कोई भी फॉरेस्ट्री स्पीशीज से, और हर ईयर वो उसके नीचे कुछ ना कुछ मेडिसिनल प्लांट कल्टीवेशन कर सकता है। तो जो टोटल बेनिफिट कॉस्ट बेनिफिट रेशियो हम लगाएंगे तो उससे फार्मर्स को मतलब वो थोड़ा सा उसको इनकम जनरेशन उसमें मिल जाएगा। और एक कॉन्सेप्ट ये हम यूज कर सकते हैं कि मतलब हमारा ये मोटिव होना चाहिए कि हाउ टू एनहांस द इनकम ऑफ फार्मर्स। अगर हम उसको सजेस्ट कर रहे हैं तो ही शुड गेट सम रिटर्न फ्रॉम इट। उसकी जो क्रॉप है इट शुड नॉट लाई इन द फील्ड इटसेल्फ।
थैंक यू मैम। एंड वन स्टूडेंट वांट टू नो दैट कि मैम कुछ ऐसे मेडिसिनल प्लांट के नाम बता दीजिए जो कैंसर के ट्रीटमेंट में यूजफुल हो सके।
जैसे टेक्सास बकाटा मैंने बताया था, टेक्सास बकाटा है जो ये उत्तराखंड हिमाचल हिल्स में होता है, तो ये कैंसर उसके लिए यूज होता है। और लोकल तो जैसे रूरल प्रैक्टिशनर्स वगैरह होते हैं तो वो लोग कई बताते हैं कि कैंसर के लिए है। तो फिलहाल तो अभी जैसे टेक्सास बकाटा मुझे ध्यान आ रहा है, एंटी कैंसर प्रॉपर्टी होती है इसका, टैक्सोल जो होता है वो एंटी कैंसरियस होता है, तो उसके लिए यूज करते हैं कैंसर उसके लिए। और एक आर्टिमीसिया करके होता है, ये भी कैंसर ट्रीटमेंट में यूज होता है।
मैम वन क्वेश्चन फ्रॉम स्टूडेंट कि जो भी आपने बताया है कि जो मेडिसिनल प्लांट है ये रियली में इफेक्टिव होते हैं या फिर ये सिर्फ मिथ है एक?
नो नो नो, दे आर रियली इफेक्टिव। आपने फॉर हम लोगों ने देख लिया है कि कोविड टाइम में हम लोगों ने ये देख लिया है कि व्हेन वी गो फॉर आयुर्वेदिक ट्रीटमेंट्स वी वर गेटिंग द सेम रिजल्ट और जब हम लोग ये एंटीबायोटिक वगैरह खा रहे हैं तो वो भी 3-4 दिन में ठीक हो रहा है, और आयुर्वेदिक जो काढ़ा पी के भी घर में रह के, तो ये तो हमारे है कि खांसी है तो हमें क्या करना है, गला खराब है तो हमें कौन सा यूज करना है। तो ये तो देखिए मॉडर्न मेडिसिन मैंने आपको दिखाया चार्ट में कि कितना कम यूज हुआ, और जो हमारा फोक मेडिसिन सिस्टम है, तो जब हम लोगों का ये हॉस्पिटल या डॉक्टर्स नहीं थे तो हम लोग इसी जो हमारे नेचुरल वेल्थ है, बायोडाइवर्सिटी है, इसी उससे हम लोग सरवाइव कर रहे थे। तो दे आर इफेक्टिव, दे आर श्योरली इफेक्टिव, नो डाउट। और अभी still if we go to rural area there are many percentage of people who are totally dependent on the plants wealth. They are not वो उन लोगों को ये नहीं मालूम कि सिरदर्द है या हड्डी टूट गई तो जाना है, दे आर टोटली डिपेंडेंट। आप अंडमान निकोबार की बात कर लीजिए तो वहां क्या ये अपने ही जो नेचुरल वेल्थ है उसमें ही पीपल आर सर्वाइविंग एंड दे आर यूजिंग दिस। So fortunate that we are rich in we have a so rich biodiversity of medicinal plant in India, especially in North India or rural areas we can say. हर एक आदमी वहाँ छोटा बच्चा भी बता देगा कि किस ट्रीटमेंट के लिए क्या यूज करना है।
मैम यहाँ पर ह्यूज क्वेश्चन है कि डायबिटीज के लिए कुछ मेडिसिनल प्लांट आप बता दीजिए।
डायबिटीज के लिए
क्योंकि
एक सिटेरिया होता है, एक सिटेरिया होता है डायबिटीज के लिए, वो है। और जामुन साइजीनियम क्यूमिनाई आप लोगों ने सुना होगा कि उसका इट इज यूजफुल फॉर डायबिटिक पीपल। और मलबरी का भी जैसे कहते हैं, मलबरी फ्रूट इज ऑल्सो गुड। और साइजीनियम क्यूमिनाई ये भी कहते हैं कि जो उसका सीड होता है अगर उसको पाउडर बना के भी खा लिया, जैसे हम लोग कहते हैं ना कि हमने जामुन खाया और सीड को फेंक देते हैं हम लोग, तो इसके लिए डायबिटीज के लिए कहा जाता है कि साइजीनियम क्यूमिनाई का अगर हम लोग वो रख लें, इसके साइजीनियम क्यूमिनाई के सीड को हम लोग मिक्सी में वो कर लें और उसके बाद खाएं, तो इट इज ऑल्सो वेरी यूजफुल फॉर डायबिटिक पीपल। और तो जैसे वो आता है इवन जैसे एक वर्नोनिया करके भी एक होता है, वो भी इट इज ऑल्सो यूजफुल इन फॉर डायबिटिक पेशेंट्स। साइजीनियम क्यूमिनाई बहुत कॉमन है।
Okay, so participants, if you have any other question, you can ask. So,
yes, sir, आलम सर कुछ पूछना चाह रहे हैं।
Yes, ma'am, your presentation is very appreciable, ma'am. I have learned a lot of things from your presentation, ma'am. I would like to ask one question from you, that if some farmers are interested in cultivating medicinal plants, so is there any agency to provide financial help to the farmer?
एग्जैक्टली जैसे नबार्ड वगैरह होता है, नबार्ड में फार्मर को नबार्ड में जा सकता है। और एक जो नेशनल मेडिसिनल प्लांट बोर्ड है न्यू दिल्ली में, उनकी एक विंग है क्वालिटी काउंसिल ऑफ इंडिया, वही लोग डील करते हैं क्वालिटी प्लांटिंग मटेरियल पर। क्वालिटी काउंसिल ऑफ इंडिया से वो जब फार्मर कांटेक्ट करेगा तो उनके बहुत सारे नॉर्म्स होते हैं, वो लोग सिर्फ क्वालिटी प्लांटिंग मटेरियल लेंगे, जैसे नेम सजेस्ट कर रहे हैं। वो अपने स्टैंडर्ड पहले से फार्मर को बता देंगे कि हम लोग किस क्वालिटी का प्लांट लेंगे। तो ये तो थोड़ा सा कॉम्प्लिकेटेड हो जाता है। नाबार्ड वगैरह फंड्स करते हैं, तो जैसे कोई एस्टैब्लिश करना चाह रहा है अपना मेडिसिनल प्लांट गार्डन या सेल पर्पस के लिए, तो नाबार्ड से फंडिंग होती है, किसानों को फंडिंग होती है। और QCI है क्वालिटी काउंसिल ऑफ इंडिया न्यू दिल्ली में, तो वो तो परचेज करता है, मतलब फंड शायद ना देता हो इसके लिए, पर वो प्लांटिंग मटेरियल किसान का परचेज करेगा, पर उनकी क्वालिटी स्टैंडर्ड को अप्रूव होना चाहिए, तब वो परचेज करते हैं QCI वाले। तो फार्मर्स कैन कॉन्टेक्ट नबार्ड, नबार्ड्स पे फंड्स होते हैं फॉर इनके लिए। अदर एजेंसीज है जैसे नेशनल मेडिसिनल प्लांट बोर्ड वगैरह, तो वो लोग जैसे फॉर एग्जांपल साइंटिस्ट या प्रोफेसर्स वगैरह को फंड देंगे। अब ये डिपेंड करता है, इट डिपेंड्स अपॉन आवर पीपल कि हम लोग अपने साथ कितने फार्मर्स को इन्वॉल्व कर सकते हैं. फंड विल बी प्रोवाइडेड टू सम परमानेंट एजेंसीज, और हम लोग अगर अपने साथ एनरोल कर ले फार्मर्स को तो हम लोग उनको अपने थ्रू फंड्स दे सकते हैं. फॉर एग्जांपल मेरे पास एक प्रोजेक्ट आया, एनएमपीबी ने फंड किया, मैंने उसमें लगा रखा है कि मुझे मेडिसिनल प्लांट का, फॉर एग्जांपल जैसे लेमन ग्रास होता है फॉर रिहैबिलिटेशन वगैरह के लिए है, ऑयल पर्पस के लिए है, फॉर मैं उसमें जैसे प्रोजेक्ट सबमिट कर दूं लेमन ग्रास के ऊपर, तो मैंने अपने साथ किसान इन्वॉल्व कर लिया और उन्होंने मुझे इतनी क्वांटिटी प्रोवाइड कर दी. तो मैंने उनको वो फाइनेंस कर दिया कि आपने इतना एरिया जोता था, इसमें ये फर्टिलाइजर लगाया था और मुझे इतना मटेरियल दे दिया. तो वो हम पे डिपेंड करेगा. एग्जैक्टली अगर किसान लोकली जाता है तो वो नाबार्ड से कांटेक्ट कर सकता है.
थैंक यू मैम, थैंक यू वेरी मच मैम.
थैंक यू.
Okay, thank you so much, ma'am. Now I would like to invite Dr. Richa Kandan, ma'am, for vote of thanks. Over to you, ma'am.
Okay, thank you, Shivam. Thank you, ma'am. On behalf of organizers, I, Dr. Richa Kandan, Department of Botany, Assistant Professor, Mrs. Khanna Girls Degree College, I would like to thank Dr. Anita Tomar ma'am for enlightening us in this very interesting topic about therapeutic potential of medicinal plants. It is really a fascinating area of research, thinking about the current scenario where herbal products have gained so much popularity during COVID-19 time. Thank you very much, ma'am, once again. We have to listen to your lecture more often in future. Thank you, ma'am. Now, I would request Dr. Sarita to continue the next session. Dr. Sarita.
Yes, ma'am. Okay. Thank you, Richa, ma'am. This is my privilege to announce the second speaker of this day, Dr. Vishnu Agrawal, Associate Professor and Head of the Department of Biotechnology, MNNIT, Prayagraj. I would request to Dr. Achila Shrivastava, ma'am, Assistant Professor, Botany Department, SS Khanna Girls Degree College, to introduce and welcome our honorable speaker. Over to you, Achila, ma'am.
Allow कर दें.
Ma'am, unmute yourself. Achila, ma'am, are you there?
उनका माइक म्यूट है क्या सरिता? शायद
हाँ,
देखिए।
माइक म्यूट है, माइक म्यूट है. अचला मैम. Just a moment. हाँ शिवम, देर इज सम टेक्निकल प्रॉब्लम. अचला मैम का माइक अनम्यूट नहीं हो रहा है. यू कैन डू समथिंग. शिवम.
Yes, ma'am, I am doing something. मैम उन्हें अपनी तरफ से ही करना पड़ेगा अनम्यूट खुद को।
Okay, then I would like to request Dr. Shabnam Parveen, Assistant Professor of Botany, SS Khanna Girls Degree College and convener of this workshop. Please make a formal welcome and introduction to Vishnu sir.
Thank you, Sarita. Today, I feel honored to have with our eminent speaker, Dr. Vishnu Agarwal, Associate Professor, Department of Biotechnology, MNIT, Allahabad. Welcome you, sir. Your presence gracing this event.
Thank you, madam.
Dr.
Thank
Vishnu
you.
Agarwal has higher education from Himachal Pradesh University and he is PhD from IIT Roorkee. He has 18 years research experience and teaching experience of 14 years. His research interest being molecular biology, microbiology, biofilm, associated infections and therapy, quorum sensing, host pathogen interaction, etc. He has produced 7 PhDs and supervising 5 PhDs currently. He has published a large number of research papers in international journals of high impact, and he has 2 patents. He is on the panel of reviewers in AICTE and members of various professional bodies such as Food Safety and Standard Authority of India, NCRT, and many more others. His academic visits include USA, France, Germany, UK, Netherlands, Hong Kong, etc. He has 5 extramural research projects funded from DST, DVT, SERB, CSIR, and ICMR. Welcome you, sir. Please, sir,
Thank you.
let us start with the presentation.
Yes, thank you very much, Dr. Shabnam. And I am particularly thankful to all organizers, Principal Professor Lalima Singh, and convener Dr. Preeti, Dr. Shabnam, Dr. Sarita, all organizing secretaries of the conference, this workshop. And the topic is very relevant, that is the green biotechnology and therapeutic potential of medicine. So may I know who are the target audience? These are UG peoples or PG or research who?
All are there.
All are there.
Yes.
Yeah. So basically now the Indian scenario has been changed a lot in terms of research, as you can see. So is my screen is sharing, right?
Yes, yes.
Yeah. So basically, I made this presentation a few days back only. Then I decided to give you a glimpse like how the things with the herbal and how the things with the research is going on. So as far as my expertise is concerned, then our group is working in the area of biofilms and quorum sensing. From since inception at MNIT Allahabad in 2009, I started my work. So basically, if I talk about the biofilm communities, so they are very simple community that are attached to the surface. They are not like other microbes. They are floating community like in water or in some simple media. But these are the surface attached communities that are responsible for a lot of infections, lot of things in its own. Suppose you traveled a lot and sometimes you complain that we have middle ear infection, like मेरे कानों में दर्द हो गया है, because आपके कान में क्या हुआ, when you open your window, the things goes inside and the microbial community develop. That is known as otitis media. That is very common. Somebody's and even the faculty members, they are using that particular lenses, and these lenses are get infected after some time. These are also, you have to change after a certain time. These are also due to the biofilms. So what are these interesting community? And these are the communities that are everywhere around you, around your wall, around your floor, around the infections, everywhere these are. So these are basically characterized by structural homogeneity and genetic diversity. So what happens, a single bacteria, they can't live alone. अकेले नहीं रहना होता है. They just make a community and this community is known as biofilm. Inside the biofilm, they produce a lot of exopolysaccharides, and under which the microbes enjoy a lot of shelter, lot of facility. Also, no immunological cell can beat in them. Okay, so very small that I know that many students who are there, like UG students, they want to know these facts. तो मैं आपको बता दूं कि whenever कोई भी microbes, any microbes enter into the body, सबसे पहले उसमें macrophage attack करते हैं. Macrophage attack करते हैं and they kill them. But these biofilms, they also have resistance towards the macrophages also. Very important communities. तो और बहुत अच्छे से हम स्टडी भी कर सकते हैं इसको लैब में. It's very simple to make. It's very good topic of research. So it's a complex aggregation of microorganism growing on a biotic or abiotic any of the surface. So these are the biofilm. So what is the difference? Like if I talking about some other system like microbes. So planktonic form of microbes, they are freely suspended and floating assemblance. But these biofilms are attached to the surface. They are generally, the normal microbes, they are generally susceptible to the antibiotics. But here in the lab, we don't have any type of susceptibility. It has is very much resistance, like 50 times resistance, 10 times resistance than the normal. तो फिर करें क्या इन बायोफिल्म का? It's a very problematic. How can we cure? How can we get a solution? In India now, everywhere people are talking about the solutions. They don't believe now on the core sciences. So what is the solution? What can be done? So with the idea, initially I started many of the research projects, funding projects, and we created some expertise giving different parts like what are biofilm. Because जब तक आप समझेंगे नहीं, you cannot give a solution to any problem. तो आपको समझना है, you need to understand. So I worked around 13-15 years on that. So what are the problems with the biofilm? So whenever we have problem, then we will contact the solution. Periodontitis, जितना भी dental cavity होते हैं, that is due to the biofilm. Otitis media, middle ear infection, heart valve infection, bone marrow infection. In other hands, all contact lenses, catheters, sutures. होते ना कि बोलते हैं आप लोग के घरों में कि अरे हम गए थे टाका लगवाया और पक गई चोट, है ना? That is sutures. They get inflamed due to the infections. Arteriovenous stents, artificial heart valves, blood vessels, they all are due to the biofilms. Who make the biofilm? The last one, the yellow one, look at these microbes, they form the biofilm. Almost 99% microbes make the biofilm. And during my PhD, I work on Candida albicans biofilm. During my even my master training was in biofilm. I was the Biblo biofilm. And now I am taking the Pseudomonas aeruginosa as a model system for the biofilm. So why biofilms are so much resistant? So a lot of research has been done on the topic, and it was found that due to the antibiotic neutralization by polysaccharide matrix, extracellular beta-lactamases are produced, and also there are some waste material are secreting that can inhibit the biofilm formation. Plasmid transfer is also a common nature. That's a horizontal gene transfer is also responsible for biofilm resistance, and also many microbes, they are also responsible for due to the resistance because they are generally in a low metabolic stage. If I ask you the difference between the microbes in normal and biofilm mode, then in normal mode the biofilms microbes are in a high metabolic state, but inside the biofilm they are in a lazy state, low metabolic state. उनको बहुत अच्छा लगता है, they enjoy to get rest, get protected, and get food, shelter, everything inside the biofilm. So this is the thing. And in the one hand, when we talk about the biofilm, then another hand, we are also having the quorum sensing. So what are the quorum sensing molecule? Basically, quorum sensing is an environmental sensing system. They allow bacteria to monitor their own population density. भाई बच्चों जो यूजी स्टूडेंट पीजी स्टूडेंट हैं, देखिए माइक्रोब्स के पास अपनी आईज नहीं है, they don't have eyes, they don't have hands. तो उनको अपने आप को देखना है कि how much we are here. तो कैसे वो सेंस करेंगे कि हम लोग कितने हो गए हैं? तो वो क्या करते हैं, खास तरह का सिग्नल सिक्रीट करते हैं. दैट सीक्रेट सिग्नल इज नोन एज कोरम सेंसिंग. तो दीज कोरम सिग्नल दे गोज आउट एंड फिर उसी को वो एक्सेप्ट करते हैं. तो जितने ज्यादा सिग्नल होंगे उतना ज्यादा उनका एक्सेप्टेंस करेंगे. फिर उनको पता चल जाता है कि ओके वी आर सो मच हियर. तो नॉट 1 microbe, not 2 microbe, not 1000 microbe, they after a certain population density they start quorum sensing and infectivity. तो ऐसा नहीं है कि अगर आपको 10 बैक्टीरिया 100 बैक्टीरिया अंदर चले जाए तो इन्फेक्शन हो ही जाए. They need a certain type of population density to get infection inside the body. So that is known as quorum sensing. And these quorum molecule, biofilm को अभी एक तरफ रख देते हैं. Now come to the quorum. These quorum molecule are of 3 types. One is in gram negative, one is gram positive, another is in both gram positive and gram negative. Give me a moment. Yes. So if I am asking about the gram positive, then it is a small oligopeptide chain that contains 2 to 20 amino acids. In gram negative, these are acyl homosyn lactone molecule, and in a AI2 signals, they are both in the gram positive and gram negative. So these 3 molecules are there. So what happens when cell density is enough, nutrition is there, environmental conditions are there, then quorum sensing starts. And what quorum sensing do? It includes bioluminescence, it includes production of antibiotics, it includes virulence factors and biofilm formation. तो अब biofilm formation यहाँ quorum से जाके correlate हो जाता है. It helps in biofilm formation also. So after a certain time, this is the flow diagram. Like microbe come, they have regulatory systems, they have chemical proportion to population chemicals for population density monitoring, they concentration threshold, then autoinducer bind to receptor in nearby bacteria, and quorum sensing activation. जैसे ही quorum sensing all of a sudden now the quorum sensing will activation will be there, then all the signaling and things will be started, and the things will be starting soon. So up we had a challenge like how to control this type of quorum molecule. Then definitely we went for some solution. What can be done? So what can be done? Definitely we have to give an therapeutic aspects. We have to go for certain therapeutics. Then we start searching the solution. The problem and challenge was that when I was going for a solution which is a synthetic component that can inhibit the molecule, again I have to go for a lot of clinical trials, lot of things, lot of other that, and compliance issues there, standardization issues there. And then we went for certain type of herbs. Okay, leave it. Then we went for certain type of herbal formulations, and we went for patenting. So 6 years back, we started working on with the solution after exploring all the pathway, all the aspects of quorum molecule and biofilm. Like my one student worked with neutrophils, one worked with the platelets, one worked with the macrophages, and after that we had all the pathway in our life explored. Abhi, what can be the particular strategy to inhibit that particular biofilm? Then we went for herbal formulations. Okay, chalo kuch start karte hain with the herbal formulation to prevent the biofilm and quorum sensing. Then the benefit of the herbal formulation over other is that it has a broad spectrum and also has a preventive medicine. It helps in rejuvenation of the entire immune system of the body. Now it has the big market, around 25,000 crore rupees market is there. Ayurvedic medicine for only Patanjali, I am saying 25,000 crore. So Ayurvedic medicine also claims it helps in treating metabolic and certain chronic condition. Many of you has got benefited also. It comes in par with the allopathic medicine and sometimes even used in challenging disease. So what can be done is, and also herbal remedies are self-contained and nutritive supplements are there. They don't have certain high side effect. And when I visited China, particular Malaysia, when I visited Hong Kong, I find that their particular things are very much, they are very advanced rather than they have a particular showrooms. Even they are working the herbal anesthesia and they are in the next level. So with the idea of that, we started working with certain type of herbal component. Okay, we can take the component. Then what can be the strategy? The challenge was that what can be strategy? It is not like that we put certain things from the here and there and just put that and make the ZOI, zone of inhibition, and started doing something. No, we cannot do that. Actually, whenever we are using certain type of component, we have to go for compliance. Okay, it should be eatable, it should be harmless, it should be everywhere readily available. Suppose if I selected some plant, मैं कोई ऐसा plant select कर लेता जो कि पूरे एक साल में एक बार उगता है, तो how can it be readily available to the all over India? So it was also a challenge. So I did a lot of research on that 6 years back, and we ultimately selected some plant. Okay, and also suppose karo if you are taking some plant, suppose turmeric le lo. So turmeric yani haldi. Simply if you're taking from the chalk or simply you're taking from the telier guns, the two varieties are different. But we have to have consistent varieties. So I put up from a certain type of exporters that they provided me with a certain certificate of analysis. Like, okay, it has certain component with that, and this much component is inside the thing. So I started work with that, and believe me, around we took around 16 different microbes from the MTCC. 16 different microbes and around 25 extracts we took. So 16 into 25, if you are going for a certain combination, then around 400, 450 combinations were there. So each time this much plates were produced. So the exhaustive work which we did, and also the MIC we did biofilm. So it took a lot of time. Ultimately, we divided our work to 4 phases. As I was asked, I asked Dr. Sarita that I will present the work, which is a very translational, and recently I filed few patents and also given technology to the market. So I will discuss. So this work I divided into 4 phases, like phase 1, phase 2, phase 3, and phase 4. And in phase 1, the screening of plant extract, it was a so exhaustive work, so much plates, like it took too much time, too much labor, MIC determination. After that, the herbal extract on biofilm. That was the phase 1. After that, when I went to the phase 2, our student, Mr. Devendra Singh, was working on that, and effect of a screen compound on bacterial quorum molecule. For quorum molecule, we have certain type of bioreporter strain in our lab. Suppose if some molecule is having quorum sensing activity, then the plate will turn white, otherwise the plate will be blue. So blue-white selection is there. We are having bioreporter strain CV026, which is TN5 mutant. So again, we decided design the formulation using experiment and Ayurvedic approach, and its analysis. How we decide? Actually, whenever suppose we have to repeat the biofilm. Biofilm has its different component. Exopolysaccharide, it's a quorum molecule, and 4-5 micros we took. Suppose karo aapko upper respiratory tract against you need a formulation. Upper respiratory tract mein many bacteria are there, like pneumonia hai, P pneumonia le lo, aureus le lo, pseudomonas le lo. Then each and every herbal extract is having different activities. So we took the best one. Again, we took the best one, which is in between the quorum sensing. Again, we took that particular thing. And in phase 3, we took, when the formulation made, we took the cytotoxicity assay, like how much cytotoxic is the formulation. in the cell line, different cell line. I have personal cell line, very good facility is there at MNIT Allahabad. So after cell line studies, we went for the brine shrimp assay also. The brine shrimp assay is a very good assay. Like is a brine shrimp is there, shrimps ke eggs hote hain. So when we put that particular egg in with the formulation, these egg cannot hatch down. उससे shrimp नहीं निकलेंगे अगर वो cytotoxic होगा. अगर cytotoxic नहीं होगा तो shrimp उसके अंदर से they will hatch out shrimps. बहुत ही wonderful assay is there. And in the after the 3 phase, these were the in vivo study. In the last phase, we went for mouse study when we confirmed, because as a part of bioethics, we cannot use the animal systems or something else in a very random way. तो ऐसे कैसे भी mouse को use नहीं किया जाता है. So we have all the legal aspects and ethical committees are there. So we go for Winster mouse we took and we go for mouse study. In the mouse, we also used the lethal dose 50, and in what doses the mouse is being killed, and after that, respect of formulation and dysentery induced mouse rat and its analysis. पहले हमने एक मॉडल लिया कि चलो उसको dysentery कराते हैं. So after that, we look at that. So these were the study. So these are the some photographs like during the screen out, I asked my student to give me some good photographs for his thesis. So these are the screening analysis, like many plant extract as giving ZOI, many are not giving, many are giving very good, many are very poor. Sometime आप लोग बोलते हो कि नीम बहुत अच्छी है, हल्दी बहुत अच्छी है, मुझे इनमें से कुछ मिला नहीं. But I got some other things in that. So we prepared a formulation for that. So you can get very good plates are there. And when I did all the work, then I also put that code word. Okay. It's a formulation 1, 2, 3, 4. That's it. Formulation 1, 2, 3, not 4. So we given the code word like what are inside, because we wanted to go for patenting of that particular formulations. So these are the biofilm formation protocol. Like just take a bacteria and put inside some microwriter plate and keep it overnight. Very simple, very user-friendly biofilm is. And after that, just rinse it and have CV0 to 6 for 2 minutes and take a OD at 590. That is the biofilm formation protocol. So this is the particular plate. Talking about the, I asked you like these mutants. When you are having the white zone, that means E component is giving quorum anti-quorum activity. Otherwise, it's not having the quorum activity. So these are the things, and this can be done. And this is the in vivo analysis which we performed at the United Institute of Pharmacy, UIP, the code number is written. Animal house is we took, and this is our rat which we given the doses, and all the doses were very good and no lethality was found. And we also treat disease rat with antibiotic, not fluoxacin, because that is the best for the dysentery. So I also do that, and treated daily for 7 days each day, and behavior and weight variation was also taken. Okay, so I found all the formulations which were the 3 formulations we tested. The best one was the formulation 1, and we took that for further analysis. So this is the mouse model. The perfect results were there. No cytotoxicity was observed. And so I got it. Okay. So based on that, I'm not going to give you more about. Yeah. So based on that, we filed 3 patents. Okay. We filed 3 patents. So number 1 is with the IIIT Allahabad. So no worries is there, no relation. But we find 3 patents. The patent 2, 3, and 4 in a screen, like antimicrobial herbal formulation, a process for preparing the same, and a biofilm anti-quorum sensing formulation and process thereof, and a formulation against UTI, United Tract, and their related bacterial infection. So, हमने ये 3 patent we have filed from MNIT Allahabad. So after that, the challenge was that, okay, patent will get granted or not granted, because these are the natural molecule. Polyherbal itself is having a lot of things. So I started working on that. Okay, patent is there, we should go for. Then patent is there, then we should go for certain type of things. So if patent is there, we have to go for technology. If something is in the market, then very good. Then I started with the companies like who want to take this formulation, which is well proven, scientific, and we published around 7-8 papers on that, whatever they were fit. And I finally transferred the technology to a new pharmaceutical player in the market, Biotechnology Pharma Private Limited. With the 2 formulation, I transferred to them to sell out in the market. So this was the overall outcome of the things, like how I started, how I made, and how I got the patent, and how I transferred to the patent. Actually, while transferring the technology, you have to go, you have to pass out from many of the things. Give me a moment, please. So the thing is that we did that. So if you want to start something, then really you need not to be a very, very do ever like atom bomb sort of things, but it's small problems can give in a better solution to the market as far as the Indian population is in the context. So these are the things, and now I would like to ask, like, if some question is there from the student side, because I would like to entertain that questions and discussions, like if they are facing something in the field.
Hello.
Hello, yes. Dr.
Yes,
Sarita,
sir.
I can speak 1 hour more, but I think it's over with the student. So it's better if they interact.
No, sir, we have enough time. If you want to say something, you can.
Actually, the thing is that when I am talking about the looking at the
Please.
college students and looking at the university systems or looking at the NIT systems, then lot of
Sir, कुछ
things.
herbal patent के बारे में students और जानना.
हाँ, herbal patent के लिए the thing is that when you go for herbal patents, then 2 things are there. देखो, एक तो होता क्या है कि you should have the proven data and proven records with you कि whatever, देखो 2 चीजें हैं, very चीजें समझ लो. One is Ayurveda, one is herbal. हो सकता है किसी कंपोनेंट का हर्बल के लिए, आयुर्वेदा के लिए पेट दर्द में रिपोर्ट हो, बट व्हेन यू आर टेस्टिंग इन द लैब यू मे गेट अ एक्टिविटी अगेंस्ट द mesenchymal cell that or some beta cell lines of pancreas, diabetic में use हो, that is your novel claim. But again, getting a herbal patent is very difficult because वो अब इतना ज्यादा saturated topic है कि working on that again you have to be scientifically proven, फिर again process फिर उसकी क्या है, you always go for the some process patent like so that is also a better thing. Yes, I think Nagma Parveen, some student has raised her hand or something. Organizers may look at.
Okay, thank you, sir, for your valuable information. It will definitely help us in future. Thank you once again. Now this is the time for more question and answers, and I would like to request Mr. Shivam Mishra to conduct this part. Over to you, Shivam.
Thank you so much, ma'am, for inviting me. Okay, thank you so much, sir, for your meaningful lecture. Sir, one question from a student that how can we cure cancer permanently? I think this is
No.
Okay, yes, sir, continue.
Actually, the thing is that this is it's not a actually these are very small molecules, many molecules, many things are involved. So the treatment of cancer is permanently, I cannot guaranteed, because it's all a matter of great, but definitely, because when I am testing, many of my colleagues are working on the different cancer, ovarian cancer, colon cancer. We have cell lines established in the lab. But when you are putting anything, even if you put some type of SARS, even they will give you activity. So it is very complex pathway. So saying like permanently can be treated, it's very difficult. हाँ, लेकिन definitely we should all like college students or university students, they should study their own model, like what can be done as SS Khanna, what can be done at NIT Allahabad. Then they should create a collaboration, and then the things can be done so that a student can also learn many things.
Okay, so thank you. So one more question. So which media to be used for quorum molecules present?
Yes, media is actually, we use bioreporter strain. So we use actually LB. LB media is there, in which we add the acyl homoserine lactone molecule from sigma aldrich. And with using the medium, it's a normal, is giving the activity. And there are specific media certain, like if you're using E. coli, then you have to add some antibiotic marker, you need to add. These type of things are there. Generally, all the work of the biofilm are done by LB and Luria Battery, agar and broth. Sometimes TSB can also be used, triptic soya broth, etc.
Okay, so and what trigger the quorum sensing?
Actually, many things triggers, like some molecules, like if a certain type of, suppose if you're working on the biosensors, then definitely certain type of enzymatic reactions, they trigger the this quorum molecule. Certain type of drugs that trigger the quorum molecule. And also bacteria itself senses, like many of your macrophages are there, they attack, then quorum molecule will be active and they deactivate the macrophages. That is the cause of infections. Yeah.
Okay, so can you please suggest some antibacterial plant, or the plant which can be used in bacterial infection?
Yeah, actually the thing is that few challenges are there. Then when people are working with the plant, many challenges are there. One is that कोई भी इतना सारा tablet तो नहीं खा सकता. Dosing is important. When I was formulating the things, तो हो क्या रहा है कि when you are putting 1 gram, then activity is given. I cannot put 1 gram because nobody can eat the 100 mg capsule, 1000 mg capsule. Paracetamol is 500. So, दो तीन चीज का ध्यान सभी को रखना है. One is that while deciding the formulation, doses देखिए. अभी आप आयुर्वेदा की दुकान में जाइए, वो आपको इतना सारा पाउडर पीस के देगा, आप खा नहीं पाएंगे. तो इम्पोर्टेंट बात है कि if we are going for a market ready technology, then we have to go for some formulations which are very less. तो प्लांट की जहां तक बात है, तो मैंने जब देखा, तो अब आप तुलसी ले लीजिए, अदरक ले लीजिए, ये हल्दी ले लीजिए, इनका इफेक्ट नहीं है, बहुत थोड़ा डालने पे इफेक्ट नहीं है. You need दूध में भी आधा चम्मच डालना पड़ेगा, then only it will help you out. When you are talking about some type of advance, then it has to be in a little quantity and go inside. Then definitely we have to go for some time of very sharp and pungent components. Like आप garlic देखिए, आप थोड़ा सा खाते हैं लेकिन आपको मतलब वो आ जाता है, इतना ज्यादा नहीं पड़ता है. तो like many of the components, if they are like some Chinese oil, are they are like thyme oil is good. Some osmum oils are there. And now I started working with some companies like Imami is also there with me. They are also giving me some things to test out. I was planning to go in their R&D, Imami Limited, जिनका Boro Plus आता है. So this is actually when you are working in a real topic, then it's very interesting to work rather than a core research. So the things is that.
Okay, so one more question. What are side effects of herbal medicines?
Herbal medicines का side effect जो होता है वो ये होता है कि suppose करो somebody is having diabetes and after taking herbal he stops his allopathic medicine, then he feels sudden sugar up down. तो धीरे धीरे इसको optimize करना चाहिए and also excess नहीं करनी चाहिए. Whatever the doses is there, whatever the things is there, and generally poly herbals का कोई side effect नहीं. Poly herbals की बात मैं कर रहा हूँ. When you go for mono herbals or some poly herbals में क्या है, देखो मैं आपको एक इंटरेस्टिंग बताता हूँ. होता क्या है कि सपोज करो तुलसी है, उसका जो एक्टिव कंपोनेंट है पॉलीफिनॉल्स वगैरह, वो जो है पॉलीफिनॉल्स वगैरह जो है, वो क्या करते हैं कि आप उनको एज सच लीव में ले लो, तुलसी की लीव खा लो, तो वो आपके लिए फायदेमंद है. बट यू टेक द प्यूरिफाइड कंपोनेंट ऑफ तुलसी पॉलीफिनॉल, इट विल हैव एलर्जिक रिएक्शन टू यू आफ्टर अ सर्टेन टाइम. to other component, they also mask the allergic activity of that particular polyherbals. तो ये एक important बात है.
Okay, sir. And so bacteriophage का use क्या bacterial disease में किया जा सकता है?
Actually, my recent ICMR project is based on that bacteriophage therapy itself. So we are looking at the macrophage cocktail. This project is going on with me. We are planning to take the bacteriophage cocktail for the biofilm related infection. And very new topic is there. BHU is also working on that. Many people are working, and definitely it has potential. If we have some, देखो पहले से लोग बोलते थे कि गंगा का पानी पियो तो सही रहते हैं. So they have bacteriophages actually. Bacteriophages का use हो सकता है, and it's very good topic, and people are funding on that. कि इसमें थोड़ा mechanism लगा के, and if some therapeutic is can be created, it's a good idea in fact. Yes.
Okay. And one more question कि bacteriophage का किस टाइप की डिजीज में यूज की bacteriophage थेरेपी का यूज किस टाइप की डिजीज में किया जा सकता है?
देखिए होता क्या है कि अभी like when you start screening the bacteriophage, then you go for some plaque formation in the plate. तो होता क्या है कि they have very specific host, like Pseudomonas के होस्ट होंगे या ओरियस के होस्ट होंगे. तो सबसे अच्छा तो मुझे ये लगता है कि सबसे पहले जो वुंड्स इन्फेक्शन होते हैं उसके ऊपर फॉज को इमोबिलाइज करके एक एंटी इन्फेक्टिव पट्टी बनाई जा सकती है कि भाई चलो एक हो गई और बढ़िया, उसमें इन्फेक्शंस नहीं होंगे. अब आप फाज को जब सेलेक्ट करते हो तो इट इज वेरी स्पेसिफिक. अगर स्यूडोमोनास सेरिजिनोसा का आपने PA001 लिया है, it is work against PA001. It's not necessary that it is working against PA14 strain. दोनों strain इंडिया में बहुत common है. तो होता क्या है कि when I am talking about the infection, then we have to screen out. But definitely, whatever the bacteria you think of, भाई Pseudomonas है, aureus है, E. coli है, and pneumonia है. For each and every bacteria, you have to isolate the FAS. फिर एक FAS की लाइब्रेरी बनानी पड़ेगी. तो उस फाज लाइब्रेरी से आप कोई भी एलिमेंट जो बैक्टीरियल डिसीज हैं वो ट्रीट हो सकता है, एंड डेफिनेटली अच्छा है. इट्स वेरी गुड टॉपिक.
Okay, so participants, if you have any questions, you can ask in chat box. Okay, sir, जब तक क्वेश्चन आ रहे हैं, क्या कुछ आप जैसे कि कुछ ऐसे प्लांट जो एंटी बैक्टीरियल की प्रॉपर्टी रखते हो या एंटी फंगल की प्रॉपर्टी रखते हो, एंटी वायरल की प्रॉपर्टी रखते हो, कुछ इसके बारे में अगर आप अपना कुछ
देखिए मैं अभी मैंने जब हर्बल शुरू किया तो उसके पीछे रीजंस ये थे कि एक तो इट्स वेरी इजी टू गेट इट्स अप्रूवल. व्हेनेवर यू गो टू द मार्केट उसका अप्रूवल नहीं मिलता है अंटिल कोई एक्ट्स हैं, लॉज हैं उनके अगेंस्ट, तो मार्केट में ही टेक्नोलॉजी रह जाती. अभी मैं ये देख रहा हूँ कि प्लांट एक्सट्रैक्ट में जैसे आप एसेंशियल ऑयल्स की बात कीजिए तो वेरी गुड एसेंशियल ऑयल्स आर हैविंग वेरी गुड एक्टिविटी. इवन आई एम वर्किंग इन एसेंशियल ऑयल ड्यू टू अभी ओमिक्रॉन की वजह से हमारा लैब बंद हो गया. अदरवाइज वी स्टार्टेड वर्किंग एंड गॉट वेरी गुड रिजल्ट्स इन एसेंशियल ऑयल. तो ये जो एसेंशियल ऑयल्स होते हैं ना, ये जो आपके मार्केट में मिलते हैं, दे आर नॉट ट्रू ऑयल्स. वो क्या करते हैं, फ्रेग्रेंस डाल के बेचते हैं. इफ यू गो फॉर सम ट्रू एसेंशियल ऑयल्स, दे आर वेरी गुड एक्टिव. इवन अभी कुछ दिनों पहले एक ऑयल का में तो हमारी प्लेट ही मतलब उसमें डिसॉल्व हो गई, इतनी अच्छी एक्टिविटी थी, प्लास्टिक ही इसमें डिसॉल्व कर दी. तो दिस ऑयल्स आर वेरी गुड एंड पीपल्स आर शुड कैच देयर एंशिएंट हर्बल मेडिसिन्स एंड एंशिएंट थिंग्स. एवरी डिसीज इज हैविंग मोर, इट नीड्स टू बी एक्सप्लोर एक्चुअली मोर. लेवेंडर ले लो, कोई भी आप ले लो, इट्स वेरी गुड.
Okay, sir. Thank you. Any other questions?
चैट बॉक्स पे कुछ क्वेश्चन आए हुए हैं.
उनके बारे में हम बात कर चुके. कुछ जैसे अभी एक क्वेश्चन है कि सर बाय ऑपरेशंस सो कैंसर विल नॉट क्योर परमानेंटली. तो क्या इसका हर्बल में कुछ है परमानेंट इसका कुछ ट्रीटमेंट?
देखिए मैं मतलब कोई मतलब ऐसा रिसर्चर नहीं हूँ मैं.
पुराने जमाने से ऐसा होता जा रहा है कि हर्ब्स में जो है बहुत पावर है, किसी भी डिजीज को ट्रीट करने की पावर होती है. तो
दैट नीड्स टू बी
कैंसर की बात कर लें,
अभी तक तो ऐसा नहीं है. दैट नीड्स टू बी एक्सप्लोर्ड. हाँ, कुछ केसेस हैं जो सही हो गए कुछ तरीके से, लेकिन अभी उसका एग्जैक्ट देखिए मैकेनिज्म में हम लोग क्यों जाते हैं, इसलिए जाते हैं कि एक बार हमें वो मैकेनिज्म पता लग जाए तो बार बार हम उसको रिपीट कर सकेंगे। तो कैसे ट्रीट हुए, उसका कोई मैकेनिज्म तो नहीं दिख रहा है, तो अभी मैं इस बारे में नहीं कह सकता कुछ। हाँ, लेकिन इन्फेक्शन ट्रीट कर सकते हैं, बायोफिल्म इन्फेक्शन ट्रीट कर सकते हैं, ये श्योर है। पेटेंट भी कर दिया है, कैप्सूल भी दे दी है मार्केट में, सून यू विल गेट इन अमेजन।
Okay, sir. Thank you so much. Now I would like to invite Dr. Durgesh Singh, sir, for vote of thanks. Over to you, sir.
Hello.
Very good afternoon to everyone present here. On the behalf of SS Khanna Girls Degree College, I, Durgesh Singh, would like to propose a deep sense of gratitude and heartily thank to Dr. Vishnu sir for accepting our invitation and giving us such a wonderful and interesting lecture about the biofilm as well as their use. And because topic of his lecture is treatment of biofilm associated infection and overview of herbal formulation and therapeutics. Thank you, sir, for your interesting and patient address. सर एक्चुअली आई वांट टू से वन थिंग कि जब हम लोग वो जो येलो कलर की आप बायोफिल्म वाली बात कर रहे थे सर,
हाँ,
वो तो जब भी हम लोग डेंटिस्ट के पास जानते हैं ना, तो ही सेड कि नहीं, ये इट इज ए coagulation of enamel, so you can remove it, it can remove by machine after
इसमें
just a minute, after removing this enamel, then they will give us an antibiotic, amoxicillin. Always they give amoxicillin for the treatment of dental problem, yeah, RCT time. So this is really very useful key. Now this this is not because of this ceremony, it is also because of some bacterial infection. So I think उनकी जो बात रही होगी और डेंटिस्ट की, वो इसीलिए amoxicillin देते रहे होंगे क्योंकि उनको पता था पहले से कि इट इज ए बैक्टीरियल इन्फेक्शन, लेकिन हम लोगों को नहीं पता था, और ये बिकॉज आपकी वजह से आज पता चला। ये पर्सनली से हम लोग बहुत मतलब थैंकफुल हैं, कम से कम इतनी छोटी छोटी डेली लाइफ की चीजें कम से कम पता चलती हैं हम लोगों को। सो थैंक यू वेरी मच सर फॉर इंटरेस्टिंग एंड यूजफुल talk. I think a student of UG and PGL as well as research scholar and some faculty member also get some new information regarding patent and all these things. So thank you sir for your kind word.
आप सबका बहुत बहुत स्वागत है ऑलवेज इन एमएनआईटी इलाहाबाद। आप लोग इसी प्रयागराज के भूमि पे रहते हैं, कभी भी यहाँ आपको कोई काम है आइए विजिट करिए। थैंक यू वेरी मच, थैंक्स फॉर ऑर्गेनाइजर्स।
Thank you.
Can I
Now,
leave now?
Yes.
Yes, sir.
Thank you.
Now I invite to Sarita ma'am for the next session.
Okay. Dear participant, this is the time for 15 minutes break. After that, we have very much interesting lecture by Navita Gupta ma'am from Jharkhand. So meeting is going on. अगर आपको जुड़े रहना है, you can join. यहीं पर आप ज्वाइन रह सकते हैं, और अगर लीव करना है तो आप लीव भी कर सकते हैं। जस्ट आफ्टर 15 मिनट फिर से लेक्चर स्टार्ट हो जाएगा। थैंक यू एवरीवन। So, Preeti ma'am, can I start again?
मैम करिए.
Okay, okay. To provide time for us in her busy schedule. Ma'am, I heartily welcome you. I would like to request Dr. College and convener of this workshop to accord a formal introduction and welcome to our honorable speaker. Over to you, Preeti ma'am.
Good afternoon to all of you. I, Dr. Preeti Singh, on behalf of the entire faculty of SS Khanna Girls Degree College, Prayagraj, cordially welcome our chairperson, Dr. Asa Seth, our principal, Professor Lalma Singh, our resource person, Dr. Navita Gupta, all the faculty members and students in this session of National Workshop. Now I feel honored to give a brief introduction of our resource person, Dr. Navita Gupta. Dr. Navita Gupta is presently working as an associate professor and head Department of Life Sciences, BBMK University, Dhanbad, Jharkhand. Her field of specialization is avian cytogenetics. In 2011, Dr. Navita Gupta was awarded a minor research project by Aero UGC Kolkata for 2 years entitled Survey and Documentation of Avifauna in Wetlands of Chota Nagpur Plateau and Bokaro District. In 2020, she has filed a patent on indigenous alcohol-based sanitizer using turmeric, aloe vera, tulsi, and neem. Dr. Navita has guided many researches and published several research papers in various national and international journals. She is an active member of WWF Bokaro chapter and director of Nature Club of India. Dr. Gupta has been nominated as a course expert group for developing pedagogically aligned executable lesson plans development by the government of Jharkhand. She has attended several seminars and workshops to update the recent developments in the area of genetics. Dr. Navita is also associated with several societies and committees. Now, without any further delay, I would like to invite our resource person, Dr. Navita Gupta, to deliver her lecture on the topic Traditional and Ancient Therapeutic Practices in Prevention and Management of COVID-19. Dr. Navita, please join us.
Thank you, Preeti, for giving
Thank
this
you.
beautiful introduction. First, I would like to thank you all the committee members, the organizing secretary of the SS Khanna Degree College. I have chosen a very layman's topic for today's lecture, that is the COVID-19 prevention and the management practices using herbal medicines. So my topic is, may I share the slide? Kindly allow me.
Yes, ma'am, sure.
Yes, ma'am, you
Okay.
are co-host. You can share.
Okay. Is it clear? Is it visible to all?
Yes, ma'am, it is visible.
Okay. So today's topic is traditional and ancient therapeutic
Yes, ma'am.
practices in prevention and management of COVID-19. So in my lecture, I have divided into two parts actually. First is what the virus actually is, the RNA virus, its mode of infection, transmission, and its pathogenicity. And in second part, I would like to add how it's managed and prevented, not cured, by using herbal medicines. So in this, so what the RNA viruses are, the coronaviruses, as we all know, these are the RNA viruses. That means the genetic component is the RNA instead of DNA, and it has name as a corona, means it has a crown-like structure on itself. As you can see here, that it has a crown-like structure on its surface. And this is coded by a protein known as a spike protein. And in addition, it has a single stranded RNA. But you see, it is a positive. Is there any problem? So it is a positively sensed single-stranded RNA, which means that, see, this is a positively strand. So it is essential here to understand what does the positively sensed single-stranded RNA means. So positively strand means it is already in the messenger RNA stage. So when it is enters into the host cell, it directly transcribes using the host ribosome. So this is due to this property, the transmission of virus into the host cell and into the community is very, very, very fast, because it is readable by the messenger ribosome directly, because it is acted as a ribosome RNA. Okay. So in addition to the RNA, it has 4 different classes of the protein. It can be a nucleocapsid protein, and it can be spike protein, envelope protein, and the membrane protein. Among all the 4 protein, the important is the spike protein. The spike protein is very important because this RNA, this positive sense single-stranded RNA, is very fast in duplication or in replication, and it can change itself. If it can change means mutation. So if this RNA mutates into a different variety, this will lead to the different form or structural form of all these 4 kinds of proteins. And among all the 4 kinds of protein, the spike protein is very important, because this actually joins with the receptors found in the respiratory tract. Basically, you see, there are 3 kinds of RNA which are causing pathogens. It can be MERS, Middle East, it can be SARS, or it can be COVID-19. So at present, these 2 forms are very much important, and COVID-19 is a novel form of SARS-CoV. So it is a SARS-CoV-2. So name. The important is that the structural difference in the core protein is important for adherence in the receptor site of the respiratory tract. For the epidemiological studies, few terms are very important, like the basic reproductive unit. This means that it is the average. See this one. It is the average number of infected contacts per infected individual. So in case of MERS, which spreads in the Middle East or in the Saudi Arabia, the value is 0.3 to 0.8. But in SARS, it increases up to 3. And in COVID-19, it's up to 2.0 to 2.5. So what is the significance of this value is, see, if the R value is greater than 1, as in case of SARS and COVID-19, that means there is a further propagation or spreading of the virus into a susceptible host. So if the R value comes to 1 and it is periodically checked, if this value is more than 1, then means the lockdown is extended, because it is still susceptible to the host. If it is less than 1, see in this case of MERS, if it is less than 1, that means the virus is extinction or doomed to extinction. So the lockdown may be relaxed here, because the spreading is now receded. So this is the meaning of the basic reproductive number. And the case fatality rate means it measures the severity of particular disease by defining the total number of deaths in a proportion of the reported cases. Means in 100 reported cases, how many death cases occurred. If you compare the case fatality rate, it is 34.4. It is less than this, and it is much less than in comparison to the MERS and the SARS. But if you compare this community attack rate, this figure is too high. This means the transmissibility of this virus is very high, which very soon spread it to the entire country except Antarctica. So the virus that spread or that starts its origin from the Wuhan city in China in 2019, and by the March 2020, it spreads to the entire continent except the Antarctica, and this led actually WHO to announce this as pandemic. So these are the comparison, and this is the novel form of the SARS-CoV, and the structural change is that this spike protein has a structure which is compatible, which fits into the receptor surfaces of the respiratory tract. Now, before going to the pandemic, or the, you can say, the curiosity among the all sections of the people across the world, that what will happen if there's a first wave, then second wave, third wave, and so on in the near future. So if a person is, say, vaccinated in the first wave, will it be effective for the second wave? Or if a person is vaccinated in the second wave, will it be effective in the third wave? This is a very important question that is a major concern for the WHO. Now you see there are two hypotheses for the spreading of the viruses and the strategies adopted for the management and the prevention. One is the antigenic drift, and second is the antigenic shift. So you see, this is a virus A, and this spreads in the first breakdown, and it has a spike protein which is coded by the RNA genome of the virus A. Suppose this virus A mutates, and you know, mutation is a random, it is a spontaneous, it may occur due to the environmental fluctuation. So suppose when it starts in the Wuhan, and some person traveling to the other parts of the world, and this virus mutates so that it has a different genome here. So this is a mutated part. And since it is a positively sensed strand, the mutation can be very easy and fast. So because of this mutation, another strain or the variant you can say here is B, which has a spike, some of the older variant and still some other. So it is a mix of the two. It may have a older, say this is the same spike protein, but has a new spike protein which is coded by the new genome. So if a person has taken vaccination in the first wave, this will remain active for the second wave also, because the spike proteins are the same, but the efficacy will be reduced. Efficacy will be reduced means maybe at the first wave, this will be a 90% efficient, and this in the second wave may get reduced to say 60%, 70%, or 75%, or it may pass or bypass the immune system. So a person, if infected in the COVID-19 in the first wave, may get infected in the second wave also. It's not mandatory that if a person is vaccinated, that means it is protected throughout the life. It's not so, because it has one type of spike of the older variant, but other is the changed one. So there is a recurrence of the symptoms in the second wave also. So this is called a genetic drift or antigenic drift. And this is happening now. We are getting too much of the variants. The genome is mutating very fast, and we getting a different types of the variants. But in other hypotheses, suppose there are two variants of the viruses. This is virus A and this is virus B actually, right? And this has a different genome. So you can see the spike proteins are different in the virus A, and in the virus B, the spike proteins is entirely different. This is because of the different genome. For some case, suppose these two genome of the virus A and B recombine or intermix, so that the third type of virus is generated with the mixed genome. If this case happens, then you can see the spike protein are totally different. Which means none of the spikes resemble either to the spikes of the virus A or to the spikes of the virus B. This is called antigenic shift. If this happens, then the pandemic will be very different. Pandemic is very different. The severity will be entirely different. There will be a drastic effect. And the WHO is concerned about this situation. What will happen if the antigenic shift is happening and the vaccination will not be affected, taken earlier in the second or in the first wave. So these are the two hypotheses of the spreading of the viruses, and it's the efficacy of the vaccines in the first wave or in the second wave. This is a major concern for the worldwide scientists. Now, since March 2020, WHO declared this as a pandemic, means it spreads across the world, excepting the Antarctica, where the temperature is too low. The WHO tracked the different types of variants that are emerged or originated in different parts of the country. And as per WHO, there are two things. One is the variant of concern, and second is the variant of interest. Among the 7 so tracked by WHO, 5 are classified as concern, variants of concern. Variants of concern because they have increase in transmissibility, they spreads very fast, increase in virulence, pathogenicity, decrease in effective public health measure or vaccination. The 5 species so identified by the WHO as variants of concern, these are alpha, beta, gamma, delta, and the Omicron, which is a hot topic of today. Now the alpha, these are the variants depending upon their genome, and these are the pango lineage system. This is a nomenclature system of the sequence of the alpha variant, and these are as B.1.17 beta variants. The beta variant has high transmissibility. This is the gamma, this is P1, delta, and this is the Omicron. And these are the earliest recorded sample in September 2020, May 2020, November 2020, October 2020, and this is the newest form called Omicron. And these are the countries from where it is recorded first and the dates. Among all the 5 variants, let me explain that the beta variants has the highest transmissibility. Now, what does the word means? Means in the alpha case, 1:3. So 1% is transmittable to a 3%, whereas in beta variant, it raise up to 1:10, which is so much of the high. But the best part is, the good part is that the vaccine which is taken in the earlier stages still remain active in the beta variant form. Omicron. Omicron is a new variant that has emerged in the multiple countries, especially in the South Africa. So what is the difference between the Delta and the Omicron? So Delta, the mutation of the Delta is almost 18 mutations recorded in the Delta as concerned to COVID-2 viruses, whereas in the Omicron, almost 43 mutations were recorded. And as a result of 18 mutations, only 8 residues are changed in the sequence of RNA, whereas in Omicron, almost 34 residues are changed. So you can see here that the Omicron is a deadly type of the variant, and Delta is that the infection or the transmission is very fast, but the recovery rates are also very good. Persons gets infected by this variant, but they are recovered very fast. Omicron, this is a type of concern that in this case, the vaccination is, or this variant is a vaccination resistant. This is the major concern among the scientists, how to tackle with this Omicron variant. 2 species are also variants of interest. These are lambda and the mu. Now, how it is transmitted? The whole story is before you. It started in the Wuhan in China in 2019, and the from which it comes through the bat. In the Wuhan city, there are many stories. It is in the market, or it comes through the, whatever the reason may be. So these are the intermediate host, or it serves as a reservoir, and the transmission among the pangolin and the bat may be a direct. But this virus mutates. Ye jo virus tha, this mutates so that the spike protein changes, and due to the structural difference of the spike protein, which is a club-shaped structure that fits very well into the ACE2 receptors of the respiratory tract. And this structural difference actually is responsible for spreading the viruses among the public domain. So this comes under the respiratory tract, and this are attached to the receptors enters into the host cell where it multiplies very fast, and very soon it expels outside through coughing or sneezing, and then there is airborne transmission, and through this there may be a direct or indirect transmission, and through this it spreads into the every part of the world, coming a pandemic type. Now this is the how it transmits. So say if this person spreads via the, so it is a zone of contact. It can be direct. It can be direct means if a person comes in range of this sneezing or the coughing, because the virus is released out of pressure along with the mucus to form air droplets. And this air droplets can spread up to less than 2 meters. So if a person comes within the range, the virus directly enters, and due to the spike, it easily anchor the body parts of this. If the droplet spreads depending upon the virus, also if the particle size is less than 5 μm, the sustain in the air for a period of time, causing the airborne transmission. There may be indirect transmission, because if a person is cough or sneezes, so it directly settle in the surfaces near to the person, and if a person sits or touches this, the virus may spread in the next victim. And for this reason, it is said that maintain 2 arms distance or 2 gaj ki duri, mask hai zaruri. And also in India, the cases, almost 20 to 30% of the cases are asymptomatic. Asymptomatic means the person is having the virus load, but the symptoms are not there. No fever, no cough, no sneezing. So person is unaware that he has infected with the virus. So wearing of mask is mandatory for all. And if the spreads is there, complete lockdown is suggested in all offices and banks, school, colleges, all are closed to break this chain of the contamination. And also the alcoholic based sanitizers are used during those days. Frequent washing of hands with soap is recommended by WHO and also by the ministry to prevent the transmission in the society. Now, this is all about what the virus is, what the mode of infection is, how it infects, how it is transmitted to the community or the society, and how it is transmitted worldwide. But what the actual disease is? See, all the viruses are mainly responsible for the cough, cold, sneezing as before, but is not deadly. But is not that deadly as the COVID-19 virus is, because it, if it affects, it will cause the severe acute respiratory syndrome or SARS, and this may include the acute respiratory distress syndrome. See, the viruses are there, it enters into nasal passage and then into the respiratory tract, where there are abundance of ACE2 receptor. It binds to the receptor and enters into the host cell, where it rapidly, very fast multiply, and then the new viral particles are expelled out through the mouth. What happens actually when it enters into the host cell? It causes systemic inflammatory reactions due to the excessive release of the pro-inflammatory cytokines and chemokines by the immune effector cells. Very sudden, these cells will come into the site or the action, and this releases the inflammatory processes, and this is called, the rapid increase is called as a cytokine storm. This is the stage. If you see the mechanism of action, this is very crucial to know how it replicates inside the host cell. As all of you know, that the virus is a non-living particle. It replicates only in the host cell, inside the cell, host cell. So suppose this is a SARS-CoV-2 virus, and it has a spike protein, which is very much fits into the ACE2. This receptor is angiotensin converting enzyme 2 receptors, and this is meant for the recepting the antibodies in the respiratory tract. But unfortunately, the receptor fits very well into this receptor cells, and this binding of this is activated by the TMPR SS2 enzyme. This is a transmembrane protein serine 2 enzyme. The presence of this enzyme activates the binding of this. So what is the difference between SARS-CoV and SARS-CoV-2? The difference is that SARS-CoV-2 has 20% high binding affinity with this receptor. इसके साथ 20% ज़्यादा था. And this is the reason it binds very fast. As it binds to this receptor, this will allow its entry into the host cell. And this is the cytoplasm part. Since it is a positive sense RNA, it does not have to depend upon the host nucleus. So it directly enters into the host cytoplasm where it has several, you see, lysosomes. And the lysosomes has the hydrolytic activity. They have protease enzyme, which dissolve the protein coat of the RNA virus. And as the protein coat is dissolved or break down, the viral genome is released free into the cytoplasm. Now comes the story that it has a positive sense strand. As it has a positive sense strand, means this act as a messenger RNA. This will act directly into the messenger RNA, and this positive sense strand of RNA finds the ribosome for the transcription process directly. So the ribosomes, which is the factory for the protein, it attaches to the ribosome and the translation of the viral protein using RNA-dependent RNA polymerase. And the RNA replicates here. Now, this is the replicated part of the RNA. You can see here, and this is the genomic replication. Since it is positive sense, the replication is very fast. It's very fast. And after this replication, there is a subgenomic which transcribes the all protein component of the RNA viruses. After transcription, the translation process goes on and the protein so formed enters into the cisternae of the endoplasmic reticulum. From endoplasmic reticulum, it enters into the Golgi complex where it is condensed, and ultimately a vesicle is formed around the virion, and this virion is expelled out through the nasal. So this is how the viral particles replicates and are expelled. But this process is very fast. This process is very fast, even faster than the bacteria replicates. It is because of the type of genome here and the less complex structure. Now comes the role of the herbal medicine. See, till date, FDA has approved no medicines for the treatment of COVID-19 virus. Only the vaccines are there, and the vaccines are the immunogenic cells. These are not for the treating of the cell. This is to improve the immunity inside the body. But the herbal medicine proves its potential in the COVID time because, and in India, there's a traditional practice of the ancient medical system. Not in India, actually, for the Asian countries. The probable reason may be because there's a biodiversity rich in the herbal plants. We have many of the documented reports in Charak Samhita, in the Pharmacopoeia, in the Chinese, Japan, and in the Nepal. The traditional practices are very popular. And say, if you have encountered cold and cough, immediately your elder will suggest kadha le lo, kadha pi lo, gargle kar lo. So all these have a proven efficacy or the potential for any kind of cough and cold or respiratory related problems. This is due. Now, so in the medicinal part, what is actually important in managing the COVID-19 or any cough, cold or immunomodulator action? So in the medicinal part, they have a specific compound called phytochemicals, or you can say secondary metabolites like tannins, saponins, alkaloid, catenins, and all. These phytochemicals or the bioactive compounds, or you can say a secondary metabolites, they have the ability to arrest the viral protection or binding or the viral replication inside the host cell. So by doing this, they will reduce the severity of the problem. But this is not that easy. If you are extracting the secondary metabolite from the medicinal plant, maybe whether traditional medicine, herbal preparation, functional food, or nutraceuticals, etc., you have to go for a clinical trials and establishment of the anticipated efficacy, and then there's a drug. So this is not an easy task, but if you review the scientific accomplishment in the year 2020 to 2021, you'll see a lot of publications as regard with the herbal extraction and its potential. Because at this time, the scientists think that this is the only way out to save drive of the management of COVID-19. Now, how it helps? Like in the earlier slide in the mechanism, we have seen that it enters into the host cell, then replicates, all the protein components are formed, then they are assembly of the viral protein, and these are expelled from the respiratory tract. At various steps, see, this is the phytochemical, this green structure. At various stages, it prevents either, number one, inhibition of viral attachment. So by taking the herbal medicine, the number one step that inhibits the viral attachment, if this step is done, that means virus is not able to penetrate inside the host cell. So if virus is not penetrate inside the host cell, there will be no replication as such, right? In second step, the medicinal property is that it also inhibits RNA replication. Say, if by some way it enters into the host cell, then this phytochemical inhibits the RNA replication, so no new virions are produced, and even if the virions are produced, they will inhibit the virus release. So these the herbal medicines have a checkpoints in the viral reproduction and the release. This is the same. Same means if you see, I'm sorry. If you see the COVID-19 disease, it can be explained in 4 stages. And in all the 4 stages, you can see these boxes explain that by taking the herbal medicine or the herbal medicaments, this will reduce the chances of getting infection or the severity of the disease. Like in the first stage, this is the before infection. Say if a person is a healthy cell, all this is cinnamon, piper, nigrum, gingiber, syzygium, all this act as an immunity booster, means the virus will not enter into the cell. So this provides the immunity boosters if you take these herbal medicines in a regular basis. Now in stage 2, say, suppose if we are taking this much of the herbal medicaments, still then the viral invades inside the host cell and the replication is going on, and the new viral particles are formed. This is stage 2. This one is a stage 2. So in this stage, if you are taking the herbal medicine like Selaginella, Allium, Agastic, and the Cucurma longa, this will inhibit the viral growth. So in the second stage, the incubation and the replication of SARS-CoV-2 is inhibited. If this inhibited, that means less of the viral particles are formed and less of the viral particles are expelled or transmitted through the airborne transmission. In the third stage, that is say, if it is replicates, if we assume that it invades and it replicates very fast, and that there is a cytochrome storm, there is cytochrome storm, then by taking the Withania somnifera, Azardica, Indica, Tinospora cordifolia, and Ocimum sanctum, if we take this medicine in a raw form or the extracts, they will act as an immunosuppressant. Means the overactivity of these cells will be inhibited at this stage. So the person may escape through the severity of the diseases, and the fatality rate may be reduced by taking this much of the herbal medicine. Now in the 4th stage, which is a deadly stage or the final stage, you can say here, by taking all these medicines, this will be post-disease stimulants, and they will likelihood that they will maybe saved because of this deadly disease. So there are 4 stages in the COVID cycle or in the virus, any virus disease, and at each step or at each stage, by taking the medicinal extract or the herbal plants, will prevent the severity of the diseases, and consequent fatal rate may be reduced. Now, this is how it checks. So you can see where the red lines are there. It checks by taking a colchicine, which is also a plant extract, this disruption of the microtubules, so that there may be no more viral trafficking and the formulation of double membrane vesicles, protease inhibit. So these are all the type of inhibition, the red blast. So this also explains the immunomodulator role of the herbal medicine or the extract, you can say. Now coming to the topic. Now, what is the role of herbal medicine or the extract in preventing the disease? These are in this figure, you can see, these are the most common ingredient of every man's kitchen. We use cinnamon, dalchini, we use turmeric, haldi, black pepper, basil, giloy, neem, garlic, ginger, and in all this, you see these have an antiviral property. So all these herbal things which are very common and which are regularly used by us, at least in the Asian people, I can say, these all have antiviral activity, antiviral potential. These are immunomodulators. They prevent the virus replication and the virus discharge or virus release. So all has and this is very known fact, not from the today's time. It is in practice from the ancient times. Like if you are experiencing cold and cough, your elder will suggest दूध में हल्दी डाल के पी लो. So this is how. So this is not a new thing for us. It is in practice from the ancient times. All the like garlic, we use garlic in the winter time much more than in the summer time. These are the things which happens. Now, these are the plant resources which are much explored during the 2020 and 2021 for its potential. And many of are very common to you. This is jamun, this is bedana, haldi, small karela, jam, and all those plants. But what is common? Why they are explored? Why they are much in the research activities? Because all of the medicinal plants are having antiviral properties. The antiviral property simply means they inhibit replication, they inhibit viral release, they inhibit the entry of the virus into the host cells. They are also immunomodulatory. The immune section, they suppress the immune cells, and they are also anti-allergy, anti-asthmatic, anti-inflammatory. So because of these properties, in the year 2020 and 2021, there are much of the publication. If you see the research activity during 2020 and 2021, you'll find a flood of the paper, not only in Asia, but also in the other foreign countries. And now we are advocating the concept of complementary and alternative medicine system. So every part of the world now is focusing on the alternative and complementary medicine system, which is an important part of the our country, because in the you see in the tribal population, many are dependent on the tribal medicines only. They never visited to the hospital for the allopathic medicines or for the surgery. They just go in the forest, pick out some of the medicinal herbs, and they consume it, and that's it. So we are already following the complementary alternative medicine since ancient times. This is the Asian medical system. So one thing is very important. When the coronavirus spreads, say in 2020, the fatality rate. The fatality rate in India is less than 1.5%, whereas in the developed countries like America, like England, like in the other countries, this is much, much higher than the recorded. This fatality rate may be, and the other fact is also that we are having a high population density. So maintaining a social distancing is just a theory, just a sentence. Practically, it is not possible in every space of the country. And so we are, so despite of the higher population density, and we are having a minimal healthcare structure, infrastructure, still then fatality rate is very low. It is less than 5%. And also we have Ayush, Ministry of Ayush, where the time to time several recommendations are sent to the nation. Use this, adopt yoga, naturopathy, kadha, and all this. So this is our part, integral part of the medicine system. What Ayush means? Ayush means Ayurveda, Yoga, Naturopathy, Unani, Siddha, and Homeopathy. This is the alternative medicine system, which we are following since long, very back. So A means Ayurveda. This is actually acronym. Y means Yoga and Naturopathy. U Unani. S stands for Siddha and the Homeopathy. And the alternative medicine system proves, their documented proof actually, that they are antimicrobial, they are antiviral, immunomodulatory, anti-allergy, anti-asthmatic, and anti-inflammatory. So by consuming the herbal medicine, I cannot say that it treats, because it never kills the virus particle, but it can reduce the severity, it can reduce the morbidity and all. So these are the formulations. Means the formulations means if you take a neem, you cannot chew the leaves straight away. So they prepare a extract, maybe mixed up with some other to make it more effective and potential. So they are herbal formulation in both, in Ayurveda, Unani, and all. The very important are, and these are very popular also. There's Kwath, Guduchi, many herbal companies prepare this one. So Guduchi, Ganavati, Agastya Haritiki, Anuthelia, this is the nasal spray. It's very effective. Unani also, Tariq-e-Raba, Arq-e-Ajiba, Khamira, and all in Siddha. So all these, they've come forward and they provide immune system very effective in the COVID period. And also, as I said, complementary and alternative medicine system, which is a integral part of the India or in Asia. And this is now a hot topic for the pharmacy company all over the world. These are the herbal formulation for the Ayurvedic. I'm not going into detail, but unique decoction or kahasya. And this is a proven fact that they had a potential of antiviral, immunomodulatory, and thrombolytic, gingival H. And during this time, you'll find a number of products mentioning this type from the Ayurvedic companies. In Patanjali, you can find a number of drugs. We can say drugs, actually, medicaments for this. And this is a gingival H tablet. This has a complex of the Phenospora, which is a giloy, and in the tablet form. And they also protects the immune system of the body. Dabur Chyawanprash is very popular among us. And since from the winter, just say winter shuru hota hai, they will suggest take Chyawanprash in your milk. So this is a proven fact that this help in the cold cough and the viral attack during the cold season. Ayurveda Advance, this is also, Ayurveda Advance is very popular during these days because it was proved that it decreased the viral load in the body by 300%. And also they help in the recovery of the patient without showing any side effect. Even then, if you find in the market, this is very, very much common, Ayurveda Advance. Ayush 64, this is the used in the influenza or in the cough and the cold. Among the Ayurvedic, these are Kavasura, Kudini, Camphor, Ayush Kwath. This is Anuthelia, nasal soap. This also help in the COVID-19 time. Among the homeopathic, not only Ayurvedic, not only Unani system, but some of the homeopathic drugs like arsenic alba. These are also antioxidant, Brania alba, Gallicinium semipervirens. These are also effective during this time and is also taken by the people time to time. Unani is less popular, but is also very much in practice during this time. And there are several common drugs during the influenza, common cold, cough, fever, and respiratory ailments like Tiryak-e-Erba, consisting of the lutea and other preparations, Khamira Marwari, and these are all very popular during the. So besides taking the Ayurveda, Unani, Siddha, and the homeopathic medicine, the two are The two are recommended during the COVID-19 time, and these are the vitamin C because of its antiviral potential, and also the zinc supplement as Zincovit. So zinc is also antiviral against viral diseases like papillomavirus, herpes virus, RSV virus, and all. And this shows its effect by inhibiting the fusion of the membrane of the host cell, decreasing the function of the viral polymerase, and blocking the release. So my aim to deliver this lecture is that the immune system should be very strong enough to fight against any dreaded viruses. And this can be done by taking the Ayurveda or any herbal medicines, which are, we are lucky that we have a diverse species, or we are, the biodiversity of the medical herbal plants are very much in the Asia as a whole. And this can be taken as the alternative medicine system till the proved medicine or the allopathic medicines come into the force. So this is how my lecture is. Now back to the Preeti ma'am.
Thank you, ma'am.
Thank you so much.
Thank you so much for your valuable information, ma'am. Now the time for question answer. I would like to invite Mr. Shivam Mishra to conduct this part of the session. Over to you, Shivam.
Thank you so much, ma'am, for inviting me. And thank you so much, ma'am, for your meaningful lecture.
Thank you.
So participants, if you have any question related to the lecture, you can ask in chat box. Ma'am, one question is there.
Yes,
as we know that COVID-19 is very infectious disease, जैसे उसके लिए 2 गज की दूरी और मास्क है जरूरी, लाइक टाइप बहुत सारी चीजें चल रही हैं. बट फैमिली में किसी एक मेंबर को अगर कोविड होता है तो जब तक हम जांच नहीं कराते हमें पता नहीं चलता कि वो कोविड पॉजिटिव है.
डेफिनेटली
उसके पहले तक हम ना तो कोई मास्क यूज करते हैं ना उनसे कोई डिस्टेंस मेंटेन करते हैं. लेकिन जब एक पर्सन का कोविड पॉजिटिव होता है और जब पूरी फैमिली को चेक कराया जाता है तो वो पॉजिटिव नहीं होते हैं, जबकि वो काफी क्लोज रहते हैं एक दूसरे के फिजिकली. उसके बावजूद ऐसा क्यों, जबकि हम जानते हैं कोविड एक बहुत ही इन्फेक्शियस डिजीज है. इस केस में ऐसा क्या हो सकता है, बाकी लोगों को क्यों नहीं हो रहा है?
सपोज कीजिए एक फैमिली में 4 लोग हैं, तो if the person is tested or not tested, there are 2 cases, either it can be tested or not tested. If a person start coughing and sneezing, immediately they have to isolate, home isolate. The one reason may be because this, जैसे मुझे ही हुआ था, मेरे घर में 5 लोग थे, जैसे ही मुझे कफ कोल्ड हुआ, immediately हम एक रूम में लॉक हो गए. Maybe because of this, I maintain my distance. So home quarantine is there. This may be the reason.
But in many cases,
why others are not tested positive.
Yes, ma'am. But in many cases की होम क्वारंटाइन भी नहीं होते हैं. जब सीरियस कंडीशन आती है तब वो जब जांच कराते हैं तो वो कोविड पॉजिटिव निकलते हैं. उसके पहले तक सारी फैमिली एक साथ रहती है. एक्चुअली मेरे नोन में काफी ऐसे केसेस हुए हैं. दैट्स व्हाई ये क्वेश्चन अराइज हुआ कि मतलब जब इतना इन्फेक्शियस है तो बाकी लोगों को क्यों नहीं हो रहा है?
Maybe the immune systems are more effective. Maybe. Like my mother is 80 plus, but she's regularly consume neem ka patta, वो जो कोमल वाले नहीं होते हैं.
Yes.
And she's more immunogenic than me, because I travel a lot. Mummy stays sit at home. वो बाहर नहीं जाती, वो बाहर का खाना नहीं खाती. She has a less chance of contact to anything you say. So maybe this be reason. But in the first wave, if I recall, if a person is tested positive, the hospital person will soon transfer that person to the hospital, and the barricade is there. आपको याद होगा, एक आती थी, खट से आके बांस बांध देती थी. And then it start contact tracing. आप पिछले 14 दिन में किस किस से मिले हो, फिर गवर्नमेंट उसके यहाँ जाती थी.
मीटिंग हिस्ट्री. यस.
इस तरीके का होता था पहले, लेकिन जब सेकंड में आया तो ये ऑलमोस्ट कम हो गया. ठीक है, बिकॉज ऑफ द वेरिएंट इज डिफरेंट. वो एक अलग सा वेरिएंट था, वहां ऑक्सीजन लेवल की प्रॉब्लम होने लगी. अब जैसे हमारे पास डेल्टा वेरिएंट है, अब हम देख रहे हैं क्या कि लोग हो तो रहे हैं, पॉजिटिव बहुत हो रहे हैं। यदि आपको थोड़ा सा भी कफ कोल्ड गए, इफ यू आर गोइंग फॉर अ टेस्ट, यू विल बी टेस्टेड पॉजिटिव डेफिनेटली 100%, बट द रिकवरी रेट इज वेरी फास्ट।
Yes, sir.
ये रिकवरी रेट क्यों फास्ट है? क्योंकि हमारी गवर्नमेंट का जो ड्राइव था, वैक्सीनेशन ड्राइव, दैट इज कमिंग इनटू, वो जो ड्राइव था उससे अभी भी उसका इफेक्टिवनेस दिख रहा है बिकॉज ऑफ द एंटीजेनिक ड्रिफ्ट।
Yes.
Suppose if there is a case of antigenic shift, all the vaccination will not work.
Okay. Thank you, ma'am.
तो इस पे लगातार बातें हो रही हैं, and every day is a new concept is coming, new theory is coming. WHO is also confused sometimes, I think so. कुछ अलग अलग वो बताते हैं जो नहीं भी
चेंज होता रहता है उनका कुछ टाइम
चेंज
पे।
होता रहता है, क्योंकि उनके मुंह में कुछ साइंटिस्ट मुंह में भर देते हैं, आप ये बोलो, तो ये सभी बातें होती हैं। ठीक है, वी आर स्टिल इन कंफ्यूजन। अभी देखिए यूरोप में कह रहे हैं कि नो नीड टू वेयर मास्क।
हाँ, अभी रिसेंट
अभी अब
उसमें
कह
आया
रहे हैं
लिव विद
अभी
कोरोना।
हाँ, लिव इट विद द सिंपली लाइक इन्फ्लुएंजा वायरस।
यस,
ये एक वायरस है, बस अपने आप को ये कीजिए। बट जब तक प्रॉपर मेडिसिन नहीं आ जाता है तब तक ये फेयर ये बना ही रहेगा। एंड यू सी इन इंडिया, कैन यू थिंक ऑफ सोशल डिस्टेंसिंग इन धारावी? डेफिनेटली नॉट।
वेरी कंजेस्टेड एरिया।
एक रूम में 4-5 लोग रहते हैं। Can you think of social distancing in that situation? If you compare the urban incidents compared to the rural is far better. वहां में incidents नहीं, वहां reported cases नहीं थे वहां पे। So this happens. कोई भी नई चीज में समय लगता ही है समझने में, और चूंकि ये जेनेटिक लीनिएजेस है, अब कहते हैं कि आर का सीक्वेंस ढूंढना चाहिए। In India, is it possible for sequencing of every patient? Definitely no.
Yes, ma'am.
That's
Thank
the
you.
reason.
I think Dr. Alam sir want to say something. Yes, sir. So please unmute yourself. Dr. Minhaz Alam sir, please unmute yourself first.
Good evening, ma'am. Navita, ma'am.
Good evening.
Myself being assistant professor of botany at Mirza Ghalib College, Gaya. So being a teacher of botany, I would like to ask from you that, ma'am, let me know whether these herbal medicines or the herbal medicines can be curative or preventive medicine in the treatment or prevention of this COVID. I think that, in my opinion, we should go for some herbal extract or herbal medicine for this, but how much it can be beneficial in treatment of this COVID? I think that it may be preventive. We can use these medicinal plants like kada and all these things to prevent the disease. But I think that it is not yet known whether it is curative or not, ma'am. So I would like from you, ma'am, to elaborate on this point.
See, medicine, if we are taking medicine, that means we are killing the pathogen. We are taking any, allopathic medicines. That means it means that it's killing the pathogen. But in case of herbal medicaments, we use the term medicaments, right? It not kill the virus. जैसे कि मैंने आपको एक स्लाइड में दिखाया था, वो वायरस को किल नहीं कर रहा है। यदि वो वायरस को किल करता तो मेडिसिन कहलाया जाता है। It is just inhibiting the entry, inhibiting the replication, inhibiting the release. So these are the immunomodulatory, we can say. This can prevent. So in my topic, it said in the prevention and management. It's not the treatment, right? Treatment means killing of the host cell. It's not killing the virus particle. It's just inhibiting, it's just suppressing. So it reduces the severity of the case. As I said, the fatality rate in India is 1.5%, where the population density is very high, where the health infrastructure is minimal. आप गांव में जाइए तो आपको कायदे का डॉक्टर नहीं मिलेगा। लेकिन हमारी fertility rate क्यों low हुई? Because we consume हल्दी on the daily basis. चाहे वो low income group का हो member या high income group का हो, हम हल्दी खाते हैं, which has an antiviral property. We consume ginger in the winter times, long, neem, and these are so. So this cannot cure. This is not a medicine. Medicine means if it kills the viral particle. यदि virus को काट मार ही दे, so there will be no symptom at all, and
Yes.
the person is said to be cured. This is not so. Right? So it
Right.
inhibits, it suppresses. This is the case. That's why it's called as a complementary and alternative medicine system.
Thank you, ma'am, for your explanation.
Thank you, and thank you, sir, for your beautiful question.
Yes.
One more question, ma'am. Can we introduce some mutagens in these viruses? One second. Yes. Can we introduce some mutagens in these viruses in positive sense of human so that they can't affect us? Is there any possibility?
See, the major mutagen, this is a positive sense single RNA, single stranded RNA. So change is very fast. And all the mutations are very random. These are spontaneous. We do not know the cause. Maybe environmental fluctuation. Like a story is there, कि person जो वहाँ से travel किया into the colder region, this mutates and this spreads in that country, and the person travel vice versa, this again comes. So what is the cause of mutation? How it mutates? And there's a theory and there's a perception that those mutations which are beneficial to that organism is that to virus will be accounted. तो अब यहाँ में क्या हो रहा है, जितनी भी mutation होता है that relates to the spike protein basically, and spike proteins are responsible for binding the receptor, and that is why this is favorable. कोई भी म्यूटेशन क्या हो सकता है, हार्मफुल भी हो सकता है या बेनिफिशियल भी हो सकता है. कोई भी ऑर्गेनिज्म उस बेनिफिशियल म्यूटेशन को कैरी ओवर करता है if it benefits the organism. So change, the structural change in the spike protein is beneficial for the virus particle as it can fit into the ACE2 receptor and enters into it. और ये चूंकि वायरस है, जब तक होस्ट के अंदर नहीं जाएगा रेप्लिकेशन नहीं होगा, इसका होस्ट तो चाहिए ही चाहिए. This is how.
Okay, again, Dr. Alam sir want to say something.
So,
Ma'am,
yeah.
I would like to know from you, ma'am, that some people say that if we wear masks, our nose should be covered. Some people use masks in such a way that their nose are open, and I think that the ACE receptor is highly concentrated in our nasal. Right. Therefore, I think that it is better to better for us to wear the mask in a better way so that our noses are not exposed. Am I correct?
No, definitely, sir. Actually, the wearing of mask correct way is to cover the nose. If you enter a public place and the guard will say, you lift your mask. यदि आप नीचे करके जाते हैं, they will say lift your mask. Because this is a fact that ACE2 receptors are also in the nasal passage.
Yes, ma'am. It is one subsidiary question, ma'am. Just I would like to know that if there is a discovery of any drug or such herbal component or any such herbal, particularly herbal or any allopathic such component which can prevent or which can reduce the binding of the receptor protein with the ACE receptor, then I think that the severity of this disease or further.
Definitely, it's going on, but you in the antigenic shift theory, this says that the mutation is in the line of modification of the spike protein that has a better binding with the ACE2 receptor. And this is the cause why we recommend the alcohol-based sanitizer.
Yes, ma'am.
So इस पे काम हो रहा है, but the thing is that the RNA mutates very fast.
Yes, ma'am.
आप 6 महीने में एक नई वेरिएंट सुन रहे हैं.
Yes, ma'am.
This may be. और ये 6 महीने में वेरिएंट हो रहा है, ये इसलिए भी हो रहा है कि स्टडीज भी कम हो रही हैं. यदि सैंपल स्टडी बढ़ा दिया जाए तो it is quite possible that you will receive a new variant within a month.
Yes, ma'am.
It is because a section of scientists are only involved in the linear study of the S gene. So if you see the articles regarding S gene, they're numerous.
Yes, ma'am. Thank you, ma'am.
Thank you, sir.
Again, thank you so much, sir, for your beautiful question. Okay, so dear participants, please on your camera for a screenshot. All participants, please.
Everyone, please switch on your video so that we can take a screenshot. After that, vote of thanks. Kindly switch on your cameras. Mute yourselves and switch on the camera only.
Okay, so now I would like to invite Dr. Achala Srivastava ma'am for vote of thanks. Over to you, ma'am. Achala ma'am. Achala ma'am, can you hear me?
अच्छा मैम अपने आप को अनम्यूट करिए.
Achla ma'am.
Achla ma'am. Connectivity problem.
Then I would like to invite Dr. Shabnam Parveen, Assistant Professor of Botany Department of this college and convener of this workshop. Ma'am, please give a vote of thanks. Shabnam ma'am.
Thank you, Sarita. Good afternoon to all of you. Myself, Dr. Shabnam Parveen, Assistant Professor, Botany Department. It is my privilege to give a vote of thanks to all those who make this event successful. On behalf of SS Khanna Girls Degree College, first of all, I want to express my heartfelt gratitude to our eminent speaker, Dr. Navita Gupta ma'am. Thank you, ma'am, for this informative and interesting lecture. I am also thankful to Dr. Anita Tomar, scientist from Forest Research Center, Rehabilitation Center, Prayagraj, and Dr. Vishnu Agarwal, Associate Professor, MNIT and Head of Biotechnology Department. I am thankful to all of you to taking out time from their busy schedule and enlightening us with their knowledgeable lecture and on this interesting topic. I am pretty sure that this precious knowledge will definitely help us in our future. Thank you so much, ma'am. I would like to also thank Dr. Asha Seth ma'am, our chairperson, our principal ma'am, Professor Lalima Singh, for their guidance and encouragement. I am thankful to all the faculty members, research scholars of different institutions of country and dear students for their kind support and patience to listening patiently this workshop. Thank you to all of you. And tomorrow we shall proceed towards the second day of workshop where we will have two interesting lectures by Dr. Sanjay Singh sir from Forest Research Center, Rehabilitation, and Dr. Lalji Singh sir from Botanical Survey of India, Andaman Nicobar region. Thank you to all of you once again. and see you tomorrow at 11 a.m. Thank you.
Thank you.
Thank you so much. And now you can lift the meeting.
Thank you.
Thank you, ma'am. Thank you, ma'am.